बोलिए, कुछ भी स्पीच करो।
Good morning. Good morning. Good morning.
I will come after 5 minutes. Okay. Thank you so much, Dr. Sanju. I'm Principal Assistant Professor. Welcome to all of you for today's International Conference. Let's make this incredible opportunity to learn, connect, and contribute to the future of agriculture. So I'm glad to invite Professor Dr. Akram Hassan, HOD Agriculture, for welcome address. Hello, phone कर। तू पहले ग्लास
Coming in the auditorium with an 3 to 4 minutes. Dear students, please be seated. Don't leave the auditorium. I request to all the students, please put your phone on silent mode.
Students, please don't leave the auditorium. Students.
We extend a very warm welcome to all the dignitaries and request them to please have your respective seats on the stage. The future of agriculture is not input intensive but knowledge intensive. Good morning, our Honorable Vice Chancellor Sir, Professor Dr. Parvinder Singh Sir, Rayat Bahara University, our today's Chief Guest Professor Dr. Ravindra Kumar Pauling, all deans, directors, authorities, faculty members, participants who have joined online and offline and dear students. I am Dr. Sanju Kumar, Assistant Professor, University School of Agricultural Sciences. On behalf of Rayat Bahara University, welcome 2 days international conference on advances in agriculture sciences organized jointly by Rayat Bahara University and Punjab State Council for Science and Technology, catalyzed by Department of Science and Technology, Government of India in collaboration with Microbiology Society India, Mohali. It's truly amazing to see so many experts, researchers, and enthusiasts from around the world in a one place to share knowledge and ideas. This conference is a platform for us to explore the latest advancements and innovations in agriculture. As we all know that agriculture plays a vital role in feeding the world's growing population while also addressing sustainability and environmental concerns. One of the most exciting aspects of this conference is the chance to connect the fellow attendees. I want to extend a warm welcome to our distinguished speakers and experts who have graciously shared their knowledge and expertise with us. Lastly, I want to express my sincere appreciations to all the sponsors, volunteers, and everyone involved in making this conference a reality. Now I request to Gurpreet for further proceedings.
Thank you so much, Dr. Sanju. I'm Gurpreet Kaur, Assistant Professor. Welcome to all of you for 2 days international conference. Let's make this incredible opportunity to learn, connect, and contribute to the future of agriculture. So I'm glad to invite Professor Dr. Amita Ma'am, HOD Agriculture, for welcome address.
Agriculture is the backbone of any stable economy, and investing in it means investing in our future. Honorable Vice Chancellor, Rayat Bahara University, Professor Dr. Ravinder Singh, Chief Guest of today's event, Honorable Vice Chancellor, MD University, Mohali, Professor Dr. Vinder K. Gori, Dr. Balbir Singh Khadda, Deputy Director, KVK Mohali, Dr. M.C. Sidhu, he is just to join within 2 minutes, and Professor Swaranjit Singh will also be joining shortly, and Dr. Maganveer Singh ji from Punjab Institute of Science and Technology. All the participants, directors, deans, HOD, faculty members of various departments, all the participants who have joined offline as well as online, and my dear students. Good morning, everyone. On behalf of Rayat Bahara University, School of Agriculture Sciences, it's my distinct
honor and privilege to extend a warm welcome to each and every one of you gathered here for the Agriculture Conference that is ICARS 2024, jointly organized by University School of Agriculture Sciences, Yadbhara University, and Punjab State Council for Science and Technology, supported and catalyzed by Department of Science and Technology, Government of India, and Microbiology Society of India. I express deep sense of gratitude to Honorable Chancellor Sir Yadbhara University, Sir Rajinder Singh Baraji, as his commitment to well-being of our students, faculty, and visionary leadership serve as an inspiration to all of us. Due to some urgent work, he could not join today. I extend heartiest welcome to Honorable Vice Chancellor Sir Yadbhara University, Professor Dr. Pravinder Singh Ji, his dedication to academic excellence, personal involvement to organize all these events, guidance and motivation behind the success of such events. My heartiest and warm welcome to Professor Dr. Ravinder Kumar Kodiji, Honorable Vice Chancellor, MD University Mohali, Founder Vice Chancellor, DAV University Jalandhar, and Central University Punjab, and moreover, he is my teacher as well as my mentor. Sir, I am so delighted to see you here as chief guest of today's event. My sincere thanks to you, sir, for spending your valuable time out of your busy schedule. I am sorry for the inconvenience caused to you. Thanks for your kind cooperation and support for that you always give to us. Welcome, sir. Welcome very much. I am obliged to Dr. Vijay Arora, Assistant Director, Punjab State Council for Science and Technology, for liberally supporting this event. My sincere thanks to Dr. Bali Bal Scientist C, who is instrumental in lending financial support to this event. Both of them could not join some urgent meeting. On behalf of Madam Jatinder Kaur, Engineer Madan B, Principal Scientist, Punjab State Council for Science and Technology has joined. Warm welcome to you, sir, for joining on this auspicious day. Warm welcome and gratitude to Dr. Baljeet Singh Khadda, Deputy Director, Krishi Vigyan Kendra Mohali. He never say no to organize any activity for our student in RBU. Thank you so very much for becoming a part of it, sir. Thank you very much. Warm welcome to Professor Faranjeet Singh Ji, but he will be joining soon. I am highly excited to witness deans, directors, HODs, participants, and dear students here for this conference. I give big welcome to all the deans, directors, HODs, participants, and dear students. I am highly excited to witness such a diverse and distinguished gathering of experts and scholars in the field of agriculture. It's a matter of pride that participants are not only from adjoining states of Punjab, Haryana, and Himachal Pradesh, but are collected from Tamil Nadu, Andhra Pradesh, Maharashtra, Madhya Pradesh, Uttar Pradesh, and so on. Agriculture is an industry that only sustains life, but also serves as the backbone of economies, cultures, and society. Today, as we continue to deliberate, exchange ideas, and forge new partnerships, we are reminded of the pivotal role agriculture plays in shaping our collective future. I would like to extend my heartfelt gratitude to all who worked tirelessly for this event. Sincere thanks to faculty of agriculture school, my colleagues of other schools, director administration, and IT team for their support, and my dear students. In closing, I wish you all the fruitful and enriching experience and ICAR 2024. May our deliberations inspire innovation, spark new ideas, and pave the way for a brighter future for agriculture. Wish you good luck and happy learning. Thank you so much.
Thank you so much, ma'am. Now I would like to invite Honorable Vice Chancellor Sir, Professor Dr. Pravinder Singh Ji, and Professor Dr. Amita Mahajan ma'am for the green welcome to all the dignitaries gathered here today. Professor Dr. Ravinder Kumar Kohli, sir. Engineer Dr. Madan B, sir. Dr. Balbir Singh Thatta.
Thank you all dignitaries. We are about to commence our event by lighting the lamp of knowledge and wisdom. This is a symbolic movement that signifies the dispelling of darkness and igniting of thoughts and enlightenment. It's our honor to invite all the dignitaries on the stage to do this honor. We deeply appreciate their presence, marking the auspicious beginning of our proceedings today.
A heartfelt thanks our esteemed guests for leading this auspicious beginning. As the lamp's light continue to radiate throughout the day, let it remind us of our collective commitment to knowledge, harmony, and progress. With this luminous start, we now officially declare the event open and proceed with our planned schedule. We are highly delighted to announce the release of conference proceedings. These proceedings encapsulated the intellectual discovery and innovative ideas that emerged during the conference. Now I request all the dignitaries kindly release the proceedings. for the dignitary. I consider it a great honor to invite Honorable Professor Dr. Pravinder Singh Sir, Vice Chancellor, Riyadh University, for word of wisdom.
Respected Chief Guest of the day, one of the wonderful personality, learned scholar, and able administrator, Professor R.K. Kohli, Vice Chancellor, MIT University, member from the Punjab State Council of Technology, Dean Academics, Professor S.K. Bansal, other deans of Riyadh University, HODs, faculty members, research scholars, dear students, ladies and gentlemen. I find it very great honor to welcome Professor R.K. Kohli, who has spent his very valuable time. A learned researcher and achiever, very upright person, man of great integrity, and above all, most disciplined teacher of Punjab University. Thank you very much, Professor. I'm thankful to Professor Amita Madan for making this event realistic despite of many hurdles in organizing this event for last 2 or 3 months. I acknowledge my gratitude to Punjab State Council for giving their financial support and other support as and when required to conduct such events to sensitize our faculty members and students in the state of Punjab. We understand that the intersection of agriculture production and research is need of our nurturing innovation and sustainable food system. We belong to a state which is agriculturally dominated, but the challenges are still very high. As I was discussing with the officers of the Punjab government, the agriculture, production and research are symbiotic elements crucial for the sustainable and advancement in the global food system. The exploration of the dynamic relationship between agriculture production, research, highlighting innovations, enhancement of the quality, productivity, these all are the challenges which higher education institutions are facing in the state of Punjab. We do understand the technology advancement in this sector is yet to take a proper momentum. Remote sensing, gene editing, revolutionary agricultural research, and the production, the benefits to the farmers of the state. The tools enable the scientists only to collect the real-time data, optimize the resources, and engineering the crops for the productivity. Thereby, efficiency and resilience in the farming system is the primary need in our Punjab state. In the contemporary era, the agricultural research encompasses the discipline including agronomy, genetics, biotechnology, and environmental science. Researchers, they are collaborating, but the farmers need to be on board. The policymakers, the industry stakeholders, they need to develop a solution for the diverse challenges such as the climate change, soil degradation, water scarcity, and the pest management. The policy frameworks is essential for fostering the enabling environment that will support the agriculture, research, innovation, sustainable production. Government must play a very crucial role in funding the research in the universities, establishing a regulatory framework, giving the incentive and adoption of the best practice of the other states and other countries of the world, and promote the inclusive agriculture policy of the Punjab state. Prioritize the small holdings of the farmers and the marginalized communities. Professor Kohli was discussing about the same issue in my office. Let's have the global collaboration in the increasing interconnected world. Collaboration among the researchers, institutions, government needs to be more concentrated with regard to the international initiatives. Research, alliance, and agriculture greenhouse gases facilitate the knowledge sharing, capacity building, coordinated efforts to tackle the global food security challenge. In the last, the important part is agriculture production research is integral component in a university that revolves around the food system and the requirement of the foods in the state. By nurturing such innovation, collaboration, sustainability, this will hold the key address in the global food security challenges. So wishing you all the best, enjoy this event and come up with the suggestions and feedback so that we can pass on the feedback to the government. Let it not be only an event of 2 days, but we should have good results from this conference, particularly from the resource persons who are expert in their field. Once again, I am grateful to Professor Kohli and all the people on the dais for coming over here and blessing our students. God bless you all. Thank you very much.
Thank you, sir, for your invaluable contributions and for being a beacon of inspiration for all of us. Now I request Professor Dr. Parvinder Singh sir and Professor Dr. Amita Mahajan ma'am to present token of appreciation to all the dignitaries. First and foremost, Professor Dr. Ravindra Kumar Kohli. And now to Engineer Dr. Magan B. sir. And now to Dr. Engineer Magan B. sir on the behalf of Dr. Jatinder Kaur Arora ma'am. Next to Dr. Balveer Singh Khatta sir. Now I request to Dr. Amita Mahajan ma'am and Dr. Magan B. sir to present token of appreciation to Professor Dr. Parvinder Singh, Vice Chancellor. Now I take great pleasure to brief about achievements of our Honorable Chief Guest Professor Dr. Ravindra Kumar Kohli, Vice Chancellor, Amity University, Mohali. Dr. R.K. Kohli is broadly accredited certified senior ecologist, decorated with several state and national level awards. He is currently the founder of Vice Chancellor of Amity University, Punjab, Mohali. He also served as Vice Chancellor of the Central University of Punjab for 6 years and as founder Vice Chancellor of the DAV University, Jalandhar for about 1.5 years. Dr. Kohli is recognized internationally for his pioneering work, especially on interplant interaction, allelopathy, and the ecology of invasive alien plants. He has successfully guided 29 MPhil and 54 PhD students, including 2 foreign students in ecology and environment. Apart from 2 patents to his credit, has published 365 research papers, authored 5 and edited 14 books. Their achievements are pretty expensive, so let me repeat. The long list of accomplishments is a testament to their unwavering dedication, exceptional talent, and relentless pursuit of excellence. Now I request Professor Dr. Ravindra Kumar Kohli to grace the stage and present the inaugural address.
Good day to you all. My younger brother, Dr. Parvinder, guests to this university for this conference, my colleagues, and dear students. I take it as a great opportunity and privilege to have been invited to this meeting conference. When Dr. Amita phoned me that we are organizing this event and we would like to, I got excited. Reason was that I wanted to again visit this campus. The difference was that earlier other people were the vice chancellors. Now my brother is there, so I wanted to see him. Thanks to the organizers for giving me this opportunity to come and share with you all my thoughts on agricultural advancements. Topic is really important, really timely. But I do not know whether whatsoever we say or we recommend, will it be applicable on the field? That is the real issue. You will agree with me that systematic agriculture started in ancient India 9,000 BCE, before the birth of Christ. 9,000 साल पहले. It was later found that later the irrigation system got developed. Initially, there used to be only one crop in a year. Then 2 harvests per year they came up. And slowly, it got commercialized as well. The later part, I am focusing more on Indian system because I do not know much about the international agreement. Then came the genetic, this genetic manipulation, hybridization started. And the role of MS Swaminathan can never be ignored when we talk about Indian agriculture. Then, of course, biotechnology. Am I audible there? It is thereafter the event of genetically engineering in biotechnology and genetic engineering. अब ठीक है? अब बताओ ठीक है? So, with the genetic engineering coming into, there were controversies, some for, some against. Leave aside those controversies. The objective is better production, Objective is quality production. Everything is targeted towards this. And when we talk of agriculture, unknowingly, we just ignore, this cattle, milk rearing or whatsoever you call it, rearing of animals and production from there. Animal husbandry, yes. Animal husbandry, we not be just ignore. It is a big mistake, in my opinion. All said and done can be improved when the agricultural land is shrinking. There has to be, for any sort of activity, there has to be a plateau. We can't go beyond a certain limit. I am an ecologist. As an ecologist, I have a different perspective, which normally in India is ignored. In foreign countries, those problems that we have here, they don't experience. That is why we have to solve our own problems ourselves. इंटेलिजेंसिया की कमी हमारे पास नहीं है. मैं उस तरह जाऊंगा कि लेक्चर के अंदर. रिसोर्सेज की कमी हमारे पास नहीं. Once when I became the DUI, Mr. Mehta, he was a correspondent. He came asking Dr. Puri, बधाई कोई थी उसने. उसके बाद कहने लगा that if you are to take birth again, where will you like to be born? मैंने कहा ये मेरे हाथ में नहीं. मुझे नहीं पता मेरे पेरेंट्स कौन होंगे. नहीं, अगर परमात्मा तुम्हें पूछे कहाँ जन्म लेना है, मैंने सीधा कहा, मैंने कहा हैरान हो गया. आपके बच्चे तो दोनों विदेश हैं. Why do you want to be born in India? मैंने कहा मुझे ये बता, इंडिया में क्या नहीं, जो विदेश में है हमारे पास वो कौन सी. मैं आपको भी गिनाता हूँ, गिनते जाना और एग्रीकल्चर के हिसाब से इवेल्युएट कर देता. हमारी सोलर पावर, किसी देश के पास you can't find so much of sunshine. Sunshine is what? It is energy. Energy is what? Energy is currency, income. Those who can spend more energy, they are richer. Those who can't, they are poor. It means we are a rich nation. Agree? We are the biggest peninsula. Peninsula, you understand? 3 sides, ocean. And we are not landlocked country. We have ocean on the borders. And we have the highest mountain peaks, Himalayas, with us. This highest peak mountains, Himalayas, they are laden with snow. Snow melts. Where does the fresh water go? It goes to the South India, to the network of rivers. और जो rivers जहाँ पर मिलती हैं समुद्र में, biggest peninsula, these coastal areas, they are the richest resources. Richest resources of minerals, richest resources of biodiversity. And all these civilizations, wherever you find in the world, जहां पर पानी है वहां पर मनुष्य आ गया, जहां पर मनुष्य आ गया है वहां पर एग्रीकल्चर होनी शुरू होगी, because man is intelligent. He knows that we have to grow food. इन साइड्स पे समुद्र हो, माउंटेन पीक्स भरी हो फ्रेश वाटर से, नेटवर्क ऑफ रिवर्स हो, और हमारी लैंड 329 मिलियन हेक्टेयर, मिलियन इज थाउजेंड, 60 लगा, मिलियन हेक्टेयर, एक हेक्टेयर 10 स्क्वायर किलोमीटर के बराबर है। उतनी arable land means there you can grow plants. Ponds को या चट्टानों को इग्नोर कर, arable land 329 million hectares. You can imagine जिस कंट्री के पास इतनी लैंड हो, इतनी सोलर पावर हो, फ्रेश वाटर हो, और हमारे सीजन्स माशाल्लाह मजा आता है। कोई भी सीजन है स्ट्रॉन्ग है, बारिशें भी आती हैं, ऐसे नहीं कि डिजल होता हो, खुला सर्दी पड़ती है तो कड़ाके की पड़ती है, गर्मी होती है तो कड़ाके की होती है। ये जो एक्सट्रीम्स ऑफ वेदर कंडीशंस, आई ट्रीट इट एज गॉड्स गिफ्ट। हमारे जितने भी बीमारियां, सब कुछ जो साइकिल है, दिस ऑल इज गवर्न बाय दीस एक्सट्रीमिटीज। किसी किसान के पास जमीन इतनी हो, पानी लगता हो, सूरज आता हो, उसको क्या चाहिए? सो किसान को मैं इंडिया मानता हूं। Then over and above human power, बंदे इसको कर्स कहते हैं कि हमारी ओवर पॉपुलेशन, मैं इसको भगवान की देन मानता हूँ, और कुछ ना मानो आप, इग्नोर एवरीथिंग, मसल पावर तो है, we can use this muscular power and treat it as a resource. कर्स कह के बात नहीं करते, और उसमें भी अगर देख लेते तभी आप गूगल सर्च करके देखो, which are the races in the world, you will find 5-6 which are considered to be intelligent races. उसमें आर्यंस का भी नाम आ जाएगा, इस एरिया के बच्चे, बार्बेरियंस का आ जाएगा, मंगोलियंस का, ग्रीक का, सबसे I mean to say that our human resource is intelligent, our human resource is hardworking, we can understand, and all those people from India who have gone abroad, 1/3 of doctors in USA are Indian doctors. 1/3 of CEOs of big companies are Indians. About 1/3 or more than 1/3 scientists working in USA are Indians. मैं अपने बच्चों को कई बार कहता हूँ कि यार अगर इंडिया वहां से निकल जाए, इसी होगा था ना, ट्रम्प ने बोला कि जी हम इम्प्लोजन होने देंगे, पॉलिटिकल गिमिक था। मैंने उसको बोला नहीं कि अब तो तू वापस आएगा ही, हम तो कहते हैं नहीं पापा वापस नहीं आने। अगर इंडियंस यहाँ से निकल गए ना तो ये कंट्री कोलैप्स हो जाएगी। Point which I am trying to write at is that our Indians, they are hardworking, they are intelligent, they can lead the world. मैं ये पॉलिटिकल भाषण नहीं दे रहा, आप एनालाइज करके देख लेना, आपको भी वहां ले जाए, you will be excelling compared to others. इन्होंने कईयों के साथ एग्जाम्पल देखा। So point is that हमारे को परमात्मा की देन बहुत ज्यादा है, हम उस देन की
tell you in the why I say so. 329 million hectares I say is our arable land. उसमें से 194 million hectares. 194 is around 60%, more than 50%. Is a degraded land. Imagine करो हमारी लैंड अगर degraded है, उस पे सॉइल नहीं है, तो इसका मतलब 50% हम लूज कर चुके हैं. जितनी मर्जी आप एग्रीकल्चर की बात कर लो, जेनेटिकली मॉडिफाइड क्रॉप्स उतनी मर्जी कर लो, कंट्रोवर्सी जो है हेल्थ इश्यूज वो चीजें लाओ ना लाओ, पर यूनिट एरिया प्रोडक्शन जितनी मर्जी बढ़ा लो, if the area is shrinking, this whole Zirakpur, ये आपका जहाँ यूनिवर्सिटी है, this whole area, हमारी जो यूनिवर्सिटी है, that area, that was all agricultural land, and this is a fertile land here. We are just ignoring the fact that we are killing our own face. हमारी requirements, we are increasing, गलत नहीं है, संबंधित को हक है, improve करने का, we must improve. But there has to be a settlement. We are crossing all that. The amount of the rate of, I should say, rate of desertification. Desertification, you understand? All those processes, all those events that create a fertile land into a desert. That is called desertification. The rate at which our Indian land is getting converted into desert, fertile land is converted into desert, is perhaps among the maximum. सबसे ज्यादा fertile land वर्ल्ड में कहीं है पता है आपको? यूक्रेन, richest, Gambia, India also is considered to be rich. Some part is of Haryana, Punjab, or UP. This area is among the rich soils. This rich soil is getting degraded. If we cannot stop this degradation, it will be irrespective whatsoever you try to do in agriculture. That will be a failure. दो चीजें हमें करनी चाहिए. And there are examples before us. One is managing water. Second is conserving soil. The fact of the matter is, Indians are not good managers. We have not been able to manage our national resources, which was our prime duty for our posterity. मतलब आप लोगों के लिए जो young boys and girls है, आप लोगों के लिए कसूरवार हम, because we have not contributed our efforts in saving those natural resources which are making us rich. Because these are natural resources given by the grace of God, we ignore. So there is always a limit. I believe that so long as we do not conserve our soil or land में फर्क है. Soil is the top crust, which is fertile. हमारे जब बारिश होती है तेज, क्योंकि soil is not covered with vegetation, that soil gets eroded. कभी आप देखें सड़कों के ऊपर जो पानी आ रहा होता है, बह रहा होता है, मिट्टी से भरा होता है, एंड आपके अपने लॉन जो हैं वो बिल्कुल साफ पानी. We are not conserving our soil, we are not putting our soil to the right type of use. And the water that erodes the fertile soil, the amount of mineral elements that get wasted to this ocean is more than the fertilizers we add into soil of our agriculture. जितनी मर्जी सब्सिडी दिए जाए, we use more than what is required. आपने कभी इजराइल की एग्रीकल्चर देखी है, कोई वीडियो देखी है? एक चीज भी, एक बूंद पानी भी वेस्ट नहीं करती। हमारे यहाँ तो पानी मिला बहुत। ये स्वामीनाथन कमीशन जब किसानों ने जो रोड ब्लॉक्स की थी, मैंने अपने ग्रुप में बात की। वैसे तो मैं जब वो मैंने कहा कहाँ खाऊंगा, मुझे कहीं नहीं लगा सरकार के खिलाफ होगा। मैंने बोला जब तक हम ये सब्सिडी कल्चर खत्म नहीं करेंगे, जो किसान मांग रहे हैं वो ठीक मांग रहे हैं, हम कोशिश कर रहे हैं कि नहीं उनको हम गोदी में उठाएंगे, गोदी में उठाने की जरूरत नहीं। He is capable. He understands much better than all of us, the scientists. उसको practical experience है। We ignore, undermine the experience of that agriculturist. जितना हम बहुत पानी दे रहे हैं, ये स्वामीनाथन कमीशन का क्यों ऐसी जगह चला? Because स्वामीनाथन had said that जितना इनपुट लगता है उतना तो उसका खर्च होगा, उसके ऊपर 50% उसको प्रॉफिट होना चाहिए। And this is justified. Plus minus बराबर कर दो, but it is justified. But इसमें पॉइंट ये आता है कि आपको पानी जो देते हैं उसकी हम कीमत नहीं देते। आपको जो सीड्स के ऊपर सब्सिडी देते हैं उसकी कीमत नहीं देते। जो फर्टिलाइजर्स आपको सब्सिडाइज देते हैं उसके ऊपर कीमत नहीं देते। आपको जो हम लोन देते हैं कम दर्जे के ऊपर लोन माफ करते हैं, you know, then political system comes to play. मैं ये मानता हूं that if we assign the right type of value to these resources, number one, we will not be wasting those resources. Number two, there will be better input to output ratio. Input मतलब कितना पैसा डाला, output कितना पैसा निकला। The nature has given us so good resources. We need to look at those resources in a more rational way. Last point is the amount of funds spent on agriculture is enough. Amount of spent on agriculture means government allocation towards the agriculture is 1 lakh and I think 30,000 crores of rupees. But I again say but towards real agricultural science is just 10% of it. Even less than 10%. Last year it was 8.3%. ये जितने PAU और IARI, HAU, पंचनगर यूनिवर्सिटी, इनके सारों के पैसे जोड़ लो, उनकी तनख्वाहें भी निकलेंगी उसी में से। और साइंस के लिए, जो रियल साइंस के लिए साइंटिस्ट के पास is very little. So long as we do not recognize their contributions, nobody else from outside is going to come and do justice for us. Our scientists are very good scientists. They understand how to take care of the land, soil, everything, and they can yield better results. But there has to be some political will. At the same time, if I were on the other side, what would I say? You know, आपको भी गवर्नमेंट में डाल दें, क्या करोगे आप? आपके पास भी लिमिटेड रिसोर्सेज, किसको कितना दूं, डिफेंस के लिए कितना दूं, इसके लिए यातायात के लिए कितना दूं और स्पेस क्राफ्ट के लिए कितना दें, हर चीजों को ये अलग अलग करना। उसको मैं ऐसे आर्गुमेंट करके डिफेंस ले लूं, but the fact of the matter is that large number of people who are non-productive, they are just getting salaries. ये कृषि भवन देख लो या किसी भी देख लो, इनके जब फंड जाते हैं फटाफट कारें खरीद लेते हैं और एसी उसी सब कुछ कर लेते हैं। मेरा झगड़ा यही होता है, डीएससी में भी यही चलता है कि भाई साइंस के लिए आप नहीं देते इतना पैसा। जो साइंस के मैनेजर्स हैं, they are making more proportionally than the real science. So, you are all my young friends. You are to grow. You are to keep in mind that so long as we do not conserve our resources, natural resources, conservation means you need to put the resource to use in such a way that it is available for use in next generation. Soil को conserve करना और water को conserve. If you have not visited Sukhumajari, कभी देखे तो नहीं रहे Sukhumajari. Request है इनको एक बार देखें. Sukhumajari is nearby. इंदौर से थोड़ा सा. That was a totally degraded land. वहाँ पर 80 families थी. 80 families में धीरे-धीरे करके सारा village को छोड़ना शुरू कर दिया. Because the land was not productive, it was or degraded land. 3 या 4 families वहाँ रह गई. यहाँ पर एक मध्य मार्ग पे there is a office of Central Soil Water and Conservation Research Institute. वो ICA आता है. वहाँ पर एक मिश्रा भी होता है, मिस्टर मिश्रा. Unmarried, about to retire. His attention was, drawn by the government of that village. कि वहाँ पर सारा village खाली हो गया. He went there. उसने villagers को कहा कि मैं आपको ठीक करना चाहता हूँ. उसकी कोई सुनता नहीं. उसने अपनी सारी सैलरी villagers को दी. क्या ये सैलरी की आपके साथ खाना खाया करूँगा. जो खिलाओगे खाओ. Villagers को उसके ऊपर कॉन्फिडेंस है. वो बेचारा पजामे कुर्ते में घूमता था, चप्पल पढ़ाई चप्पल पहन के चलता था. Villagers को उसके ऊपर विश्वास हुआ कि ये करना था. उसने सबसे पहला एक काम किया कि उनके जितने भी कैटल थी, बकरियां थी, उनको स्टॉल फीडिंग शुरू करवा दी. ये बंधी रहेंगी. We will cut the grass, cut the vegetation, and feed them at their place. So that वो जाकर और जब गाय भैंस जाती है, पौधे उखाड़ती है तो मिट्टी की पड़ती है. तो उसने एक काम किया सबसे पहले. सेकेंड, he identified an area जिसमें उसने खड्डा खोद के बहुत बड़ा पॉन्ड बनाने के लिए उसकी मिट्टी ऊपर करके और रास्ता आने जाने का बना दिया. विलेज का सारा पानी उस पॉन्ड में आता. पॉन्ड के नीचे उसने पाइप लगाकर एक पंप लगाया जिससे एग्रीकल्चर फील्ड को पानी चला. और जो रास्ता था, ये जो आता है ना हमारे सुखना लेक में पहले काफी ज्यादा सिल था. उस एरिया से आती बैटल की कैचमेंट थी. उसने बीच में बी नॉट्स बना दी. कुछ फोल्डर रखती है, पत्थर रखती है ताकि पानी का फ्लो धीरे हो जाए. जहाँ पर बी नॉट्स बना है, पानी आएगा यहाँ, मिट्टी वाला मिट्टी यहाँ रह जाएगी, पानी ऊपर से फिल्टर होकर आगे चला जाएगा, उसकी स्पीड कम हो जाए. ऐसे उसने कई ऐसे इंटरवेंशंस करके और वहां की सॉइल को, फर्टाइल सॉइल को पानी को रोक लिया, फर्टाइल सॉइल को रोक लिया. एग्रीकल्चर फील्ड्स में लोग आ गया. खेती उनकी बहुत अच्छी हुई. जितने बंदे बाहर गए थे विलेज छोड़ के सब वापस. इट इज ए गुड एग्जाम्पल. वहां पर उनके साथ होते थे डॉक्टर गरेवाल यहाँ भी आए थे आपके घर. So he is on the professor. So उन्होंने वर्ल्ड बैंक के कई ऐसे प्रोजेक्ट किए हैं. He is known for conserving the soil and water. So my request to you is to go and see there and start imagining, start thinking how to find solutions to your area that the rich soil does not go down the drain. So if you are able to do it, हम जितनी ये organic crop उधर में गया नहीं जाके और genetic engineering की तरफ नहीं गया, because I wanted, this is the basic issue. So long as this basic issue is not understood, you will not be able to achieve any good results. So with these words, I pray to the Almighty to give success to this event of International Conference on Agriculture, advancement in agriculture, and wish for you good health, happiness, success in life. Thank you so much. God bless.
Round of applause for Professor Dr. Vinder Kumar Kohli for setting the stage and eloquently outlining the purpose of our gathering. Thank you so much, sir. Due to certain other engagements, our honorable guest of honor, Professor Dr. Tinder Kaur Arora, Executive Director, Punjab State Council for Science and Technology, is unable to join us on this occasion. Madam, thank you so much for your kind and liberal support for this event. So Dr. Madanveer, Scientist, Punjab State Council, has joined on behalf of her. Dr. Madanveer Singh Ji is working as Principal Scientific Officer with Punjab State Council for Science and Technology since last 17 years. He is working towards addressing technological gaps of industry with respect to air pollution control, energy efficiency and products, and process improvement and introduction of green and clean fuels. So he has lengthy list of accomplishments. So I will now I invite to Dr. Madanveer sir for sharing his valuable words.
A very good afternoon to the other dignitaries on the dais, to the participants, to the faculty and students of Riyadh Bala University. Madam Chair, I am Madanveer Singh. I am working as Principal Scientific Officer with Punjab State Council for Science and Technology. Our organization is the think tank of Government of Punjab on issues with regards to science, technology, and environment. We are a team of 25 dedicated engineers and scientists from different sectors. We are aggressively and extensively working on addressing issues related to environmental and social grand challenges of the state, and also addressing the grand challenges of the micro and small scale industry, as well as the primary and agriculture sector. So we have shared that we are providing guidance, technical guidance to industry with regards to local industry, with regards to air pollution control, energy efficiency, shift from solid fuel like fossil fuel coal to gaseous fuel, PNG, and green fuel that may be petroleum-based products. So in the last 7 years, we have provided technical guidance to more than 1,700 industries, which has led to reduction in 50,000 tons of SPM emissions, dust emissions per annum, reduction in coal consumption by 1.25 lakh tons per annum, reduction in greenhouse gas emissions by 3.25 lakh tons per annum, and the savings of about 450 crores through investment of 750 crores in cleaner technology. That is the payback of that. So I am also looking after the climate change knowledge center, and we have recently prepared the state action plan on climate change, which covers agriculture sector and other various sectors. And we are one of the partners in this two-day conference. So and agriculture as sir has shared the background of the agriculture. So I would like to add on it, like sir shared that in now 9,000 to 10,000 BC, agriculture was the thing that civilized the humans. Before that, we were hunters and gatherers. And you can say the first thing we started harvesting was wheat. So we can say wheat is the seed of human civilization. Wheat came to the humans basically. So we started harvesting it, collecting it, storing it for the times of droughts and famines. So, and if
if we jump, so after that, if we jump into mid 20th century, 1950s, what we were staring at, big population growth. So in 1968, a US biologist, Paul Ehrlich, in his book Population Bomb, said that in coming one decade, millions of 100 million people would die out across the world because of starvation, and India would not be able to serve its 200 additional million population additional that would be in next one decade. But he didn't accounted for was contribution of agriculture scientists. In 1970, agriculture, one of my favorite heroes is Norman Borlaug. आपने सबने नाम सुना होगा उनका, एग्रीकल्चर में, पर ये दुख ये है कि एग्रीकल्चर के बाद बहुत कम लोगों को इनका नाम पता है. He developed the high yielding wheat variety, after which maize came up and rice came up. And with the development of this hybrid variety and with the use of synthetic fertilizers developed by Weber and Bosch, we could increase the per acre yield from 500 kg per hectare to 5,000 kg per hectare. So now what we are looking at, the issue, so you can say the negativity, the pessimism about the population was addressed. Now now India produces about 3 times of its annual food grain requirement. But with the use of rampant use of chemical fertilizers and chemical pesticides, what we are looking at it is degrading our soil, our health, our water, it's polluting our air. So what can we do to address this? So this is the question for the agriculture scientists, that what are the issues with this agriculture that we are currently facing and how can we address it? I'll just discuss the 4 main issues which we think are the current challenges what we are facing. First is how to develop biofertilizers and biochemical and biopesticides without impacting the yield of the crop. So to ensure food safety. Second, the agriculture sector is said to be the largest contributor to GHG emissions. Globally, we emit 52 billion tons of GHG, and agriculture sector alone emits 22% of GHG. And out of 22% of GHG, major 80% portion is from non-CO2 greenhouse gases. And this is I'm talking about the agriculture and allied sector, livestock and forestry. So this is the scenario of the GHG emissions, and the CO2 emissions basically comes from the use of tractors and combines and harvesting, cultivation, and operation of pumps for diesel pumps and electric pumps. And the non-CO2 emissions come from anaerobic fermentation in livestock, from manure, from paddy cultivation. That is mainly methane. Methane has a global warming potential of 28 times of CO2. And then major contributor to GHG emission is the release of nitrous oxide from the use of chemical fertilizers. When we apply chemical fertilizers, 50% is absorbed by the plant and the remaining is run off to the groundwater, the water bodies, and the other is converted to nitrous oxide, which has a global warming potential of 265 times. So we need to find solutions to reduce GHG emissions from this sector, because the main, you can say, climate change and also hit to the agriculture sector. It is now established that increase in 1 degree of global temperature would be detrimental to the yield of wheat as well as other crops. So some solution that will be lower heavy. Handling the large amount of agriculture. As we are saying, seeing Punjab alone produces 20 million tons of extra because of its properties of high silica content, alkaline earth metals. It is not being able to use as animal fodder. We are trying, this government has and the central and state government has put in enormous effort to utilize this enormous amount of extra, but still almost 20-15% is still be burnt in open space. So we need to find techno-economical feasible solutions to address this problem. And last is, as I discussed, that we need technologies and low-cost technologies to solve the problems of the small and marginal process. So now what we require is a multi-sectoral approach, not of the agriculture scientists, of the electronic engineers, of the we need to use AI, big data to solve these problems. So it is I urge the upcoming and budding climate scientists to come up and address these challenges. Thank you.
Thank you, Engineer Magadeep Singh, sir, for illuminating our minds with your thoughts. We are grateful that you share your expertise and enthusiastically engage our audience. The ultimate goal of farming is not growing a crop, but the cultivation and perfection of human beings. A very heartiest good afternoon to you all. My name is Dayami, and it's my absolute pleasure to extend my heartiest and warm welcome to each one of you present here at the International Conference on Advances in Agricultural Sciences 2024. First and foremost, I would like to extend a cordial welcome to our next keynote speaker, Dr. Balbir Singh Khadda, sir, Deputy Director, Krishi Vigyan Kendra, Mohali. He has been exclusively working on different aspects of animal husbandry from the past 22 years. He has remarkable and notable contribution in publication of 84 research articles in National Journal of Agriculture. I would like to request him to please come and enlighten all of us with his knowledge. Everyone, please give a big round of applause for Dr. Balbir Singh Khadda, sir.
स्टूडेंट वीरो, थोड़ी देर बैठिए, है ना? मैं आपको ज्यादा पकाऊंगा नहीं, लेकिन बड़े काम की चीज है, उसके ऊपर बड़ा सिंपल सा टॉपिक, उसके ऊपर थोड़ा डिस्कशन करेंगे, लेकिन अगर आप प्रेम से सुनेंगे तो एक्चुअल में मेरे को स्टूडेंटों के साथ में इंटरेक्ट होने में बड़ा मजा आता है, क्योंकि मैं अभी अपने आप को स्टूडेंट ही समझता हूँ। एमपीएससी, पीएचडी सभी की क्लासें लेने का भी मौका मिलता है मेरे को। गुरु अंगद देव वेटनरी एनिमल साइंस यूनिवर्सिटी है लुधियाना, वहां पे। इससे पहले मैं पंतनगर के अंदर भी 3 साल स्टूडेंटों के साथ इंटरेक्ट किया है। आज का जो मेरा टॉपिक है वो है प्रेजेंट स्टेटस ऑफ गोट फार्मिंग इन इंडिया। ये स्पेशली स्टूडेंट्स के लिए है। आजकल हर चीज का एक बूम आता है ना, कि ये कर लो, ये हो जाएगा एक दिन के अंदर। खास तौर से आपका जो यूट्यूब चैनल, सोशल मीडिया, इसके अंदर भी बड़ी चीजें दिखाते हैं, करोड़पति बना देते हैं रातों रात। तो बड़े सिंपल के अंदर अपने बात करना चाहते हैं। इसको फुल स्क्रीन कर देंगे ये क्या? कहाँ से होगा? नेक्स्ट करिए। सबसे पहले एक जो मिथ है वो मैं आपको बता दूं। आमतौर पे बकरी को पहले अच्छे घरों में नहीं पालते थे। मानते हो इस बात को? कि ये तो बिल्कुल गरीब आदमी का पशु है, वो ही इसको पालेंगे। अगर यहाँ किसी सरदार के घर पे जाओगे हो, ये कहते बकरी ओए सीधा मर्जा पालेंगे, है ना? तो बकरी साडा काम थोड़ा ही है। बकरी चंगी नहीं होती है, बकरी का दूध चंगा नहीं होता है। ये कंप्लीटली रॉन्ग। सबसे पहली चीज, इस दुनिया में डोमेस्टिकेट जब एनिमल का किया गया, या जो पशु इंसानों के साथ, जिस पशु ने रहना स्टार्ट किया, डॉग के बाद बकरी ही थी। पहली चीज, मेरा इस बकरी के साथ में, हालांकि मैं बचपन में मेरे को बकरी से बहुत नफरत थी, जो मैंने अभी आपको बताया कि मेरे घर पे भी यही कंडीशन थी, वो बकरी। लेकिन कहते हैं कि प्रकृति में जिस चीज से ज्यादा भगते हैं उसी चीज के साथ आपका इंटरेक्ट होता है। और मेरे को जब साइंटिस्ट की नौकरी मिली बकरी में, तो मैंने अपने जो सबसे सुनहरे पल थे वो मैंने बकरियों के साथ ही गुजारे, मैं और मेरी बकरियां। जिस चीज को मैंने सबसे ज्यादा फील किया, जैसे हर घर के अंदर डॉगी होता है ना, बड़ा इंटरेक्ट करता है मालिक से, सेम सिचुएशन बकरी की है। अपने मालिक के साथ वो इतना प्रेम से रहती है, जब भी वो आता है वो बहुत खुश होती है। दूसरी चीज, दुनिया की सबसे सफाई प्रिय अगर कोई एनिमल है वो बकरी है। अगर इसका आपको देखना चाहते हो, कहीं भी चले जाना, बकरी कभी भी गंदी जगह पे नहीं बैठेगी, वो एकदम साफ सफाई की जगह बैठेगी। अगर उसको जगह नहीं मिल रही है, जैसे सपोज करिए यहाँ नीचे ला के रख दिया आपने किसी बकरी को या बच्ची को, वो सीधा जाके चेयर पे बैठेगी या उस टेबल पे बैठेगी, वो नीचे नहीं बैठेगी। बकरी एक ऐसा जानवर है जो कभी भी नीचे गिरा हुआ खाना नहीं खाती, हमेशा ऊपर की टहनियां है उसी की वो ग्रेजिंग करती है। अगर आपने नीचे उसको मेंजर में डाल दिया है, वो बकरी नहीं खाएगी। उसके अलावा बात आते हैं अपने, इवन मैंने तो यहां तक देखा है कि कई ग्रुप के अंदर भैंस के साथ में लोगों ने बकरी रख रखी थी, तो जहां गोबर भी है पेशाब भी करती है, लेकिन बकरी भैंस के ऊपर बैठती है, क्योंकि एकदम मेरा जल्दबाजी के अंदर मैं आउट ऑफ स्टेशन गया हुआ था प्रेजेंटेशन में, नहीं तो मैं आपको बड़े अच्छे अच्छे स्टेज दिखाता बकरियों के। ठीक है, अब आती है दूध की बात। इस दुनिया में, इस प्रकृति में माँ के दूध के बाद अगर किसी का दूध सबसे हेल्थी है बच्चे के लिए, बूढ़े के लिए, वो बकरी का है। और इस चीज को आप सभी जानते होंगे, जिन माओं के अगर दूध कम बनता है या कोई एक्सीडेंटल दूध नहीं मिलता बच्चे को, तो उस बच्चे के लिए बकरी का दूध लाया जाता है। ठीक है, उसका रीजन ये है कि उसके अंदर जो फैट्स के जो ग्लोब्स की आकार होती है वो सबसे कम होती है, इजी डाइजेशन, चाहे वो बच्चे के लिए हो, बुजुर्गों के लिए हो। और अभी लेटेस्ट में आपने पिछले 2-4 साल के अंदर अगर सुना है, तो सबसे ज्यादा अगर प्लेटलेट्स बढ़ाती है वो बकरी का दूध है, इवन 1000 रुपए किलो तक भी गया है। ठीक है, तो ये तो बेसिक चीज है बकरी की। अब जैसे बात करें, मैंने पहले बता दिया, और पूरे वर्ल्ड दुनिया का ऐसा कोई भी कोना नहीं है जहां बकरी का पालन नहीं किया जाता है। ठीक है, नेक्स्ट। ये थोड़ा सा बेसिक चीजें हैं कि बकरियों का जो ब्लॉक की साइज है वो 2 बकरी से लेके और 3000 बकरियां तक लोगों के पास ब्लॉक है। आउट ऑफ 95% जो मेल किड्स है वो सेल कर दिए जाते हैं 3 से 6 महीने की। ये बेसिक फैक्ट है। टोटल इंडिया के अंदर अगर अपने पॉपुलेशन की बात करें तो 148.88 मिलियन गोट है। ठीक है, उसमें से सिर्फ 1 या 1.5% ही ऑर्गेनाइज्ड है, बाकी ज्यादातर स्मॉल लैंडलेस या जो गरीब तबका है उन्हीं के द्वारा पाली जाती है। अब वो क्यों पालते हैं उसका भी रीजन बता दूं मैं। एक भैंस लेके आते हो, 1 लाख रुपए का खर्चा आता है पहले परचेज करने में, और उसके बाद जो उसका डेली का जो कॉस्ट ऑफ प्रोडक्शन है दूध की, परजेंटली 30 रुपए के करीब आती है। अगर पर अपने कॉस्ट ऑफ कल्टिव इसकी निकाले प्रोडक्शन दूध की, तो 27 से 30 रुपए। उसी 1 लाख रुपए के अंदर आप 10 बकरी पाल सकते हो और आराम से अपना एक परिवार रोजी रोटी कमा सकता है। ज्यादातर जो बकरी है जो लेडीज है, वुमेन है, उनके द्वारा हैंडल की जाती है, उनके द्वारा उनकी मैनेजमेंट किया जाता है। इसलिए जो लेडीज ग्रुप है उसका सबसे बड़ा इसके अंदर योगदान है। वेटनरी की जो फैसिलिटी है वो गोट के लिए नाम बराबर है। आप किसी भी गांव में वेटनरियन के पास जाइए, डॉक्टर के पास, आपको नहीं मिलेगा। वो कहेगा जी गाय भैंस का भी इलाज कर देता हूँ, लेकिन बकरी का क्या लगेगा डॉक्टर, ज्यादा ध्यान नहीं। उसका रीजन ये है कि गाय भैंस के लिए आपको तुरंत 1000 रुपए निकाल के धनी दे देगा, लेकिन अगर इसके लिए 50 रुपए मांगते हो तो वो थोड़ा आनाकानी करता है। ठीक है, नेक्स्ट, ये जो एक गोट के बेसिक नॉलेज है, खासतौर से स्टूडेंट्स, ये पूरी लाइफ आपके काम आएगा। सेक्सुअल जो मेच्योरिटी आती है फीमेल के अंदर 6 से 10 मंथ के अंदर आ जाती है, और वही अगर कैटल एंड बुफेलो की हम बात करते हैं तो 3.5-4 साल लग जाते हैं। एज फॉर बेस्ट ब्रीड फीमेल को करते हैं तो 15 से 18 मंथ की एज के अंदर कर सकते हैं, और जो ब्रीडिंग के लिए मेल यूज करते हैं वो 18 से 24 यानी 1.5 से 2 साल का जब मेल होता है उसको यूज कर सकते हो आप। एस्टर 18 से 36 घंटे। अब ये थोड़ा सा टिपिकली है, लेकिन स्टूडेंटों के लिए है। बेसिक चीज ये है कि ये ओस्टस क्या है, यानी हीट कितने टाइम रहती है, वो रहती है 18 से 36 घंटे। उसके बाद आता है ओस्टस साइकिल। अगर एक बार हीट चूक गए तो वापस वो 17 से 21 दिन यानी एक्चुअल के अंदर 20 एक दिन के एवरेज के अंदर आ जाती है ये। एज एट फर्स्ट किडिंग 15 से 25। जेस्टेशन पीरियड की बात करें, जेस्टेशन पीरियड की अगर हम बात करते हैं तो बड़ा सिंपल सा एक तरीका बताता हूं मैं। वुमेन में कितने हैं सबको पता होगा ना, 9 महीने। 9 महीने माँ अपने पेट में बच्चा रखती है, ठीक है। गाय के अंदर 10वें महीने में, भैंस के अंदर 11वां महीना लगते हैं। कैमल की अगर हम बात करें तो 13 महीने, और अगर सीप एंड गोट की बात करते हैं तो 5 महीने। यानी आधे टाइम के अंदर ये 2 बार बच्चे दे देती है, और एवरेज जो किडिंग साइज होती है वो 1.5 यानी 1.5 बच्चे एवरेज जो मानते हैं। तो यानी अगर 1 साल के अंदर आपको 3 बच्चे प्रोवाइड करवा सकती है, ठीक है। और नेक्स्ट, अब बात आती है जो बकरी पालनी क्यों। Goats regards as ATM, anytime money, anytime milk, anytime marketing। जब चाहो तब दूध निकाल सकते हो, ठीक है। मल्टी फंक्शनल एनिमल है, ये मिल्क के लिए भी है, स्किन के लिए, मैन्योरिंग के लिए, और प्रॉफिटेबल एंटरप्राइजेज है। यानी इससे बड़ा लाभदायक कोई धंधा नहीं है। हर साल एफडी पकाती है। बैंक में एफडी कितने साल से पकती है पता है स्टूडेंट्स, आइडिया कुछ? शायद 10 से 12 साल लगते हैं आजकल। और ये पालन जिसके अंदर आपको हर साल एफडी मैच्योर करती है। 1000 रुपए का इन्वेस्टमेंट किया है तो 1 साल के बाद आपको 2000 रुपए आपको मिल जाएंगे, ठीक है। प्रोलिफिक ब्रीडर का मतलब है पर किडिंग कितने ज्यादा से ज्यादा बच्चे दे सकती है, ठीक है। पुअर्समैन काउ, गरीब की गाय है, चलता फिरता फ्रीज है, और क्विक रिटर्न, तुरंत आपको उसका रिटर्न मिल जाता है। नेक्स्ट, दूध का मैंने पहले ही बता दिया। चीवन जो इसका मीट होता है उसके ऊपर किसी भी धर्म का कोई पाबंदी नहीं है, सभी धर्म वाले बड़े आराम से खाते हैं, ठीक है। और वुमेन इंपावरमेंट है, लो इन्वेस्टमेंट है, इनिशियल जो भी आपका इन्वेस्टमेंट है वो लो है। अब नेक्स्ट चलिए, ये पूरे वर्ल्ड की गोट पॉपुलेशन है, जिसके अंदर सबसे ज्यादा गोट की पॉपुलेशन है वो एशिया के अंदर है, अप्रोक्सीमेटली 58% के अंदर। और एशिया के अंदर सबसे ज्यादा अगर अपनी बात करें इंडिया के अंदर, तो इंडिया के अंदर 148 के करीब है, और पूरे वर्ल्ड की 13% के करीब अपने यहाँ पे पॉपुलेशन इंडिया के अंदर है। नेक्स्ट, ब्रीड की बात करते हैं तो ब्रीड अपने 37 वेल रिकॉग्नाइज्ड जो रजिस्टर्ड ब्रीड है कोर्ट की वो है। पंजाब के अगर स्टूडेंट ज्यादा है तो पंजाब वालों के लिए मैं बता दूं बीटल इज द बेस्ट, ठीक है। नेक्स्ट, ये अगर दूध की बात करें तो एशिया जो है उसी के अंदर सबसे ज्यादा मिल्क का, सबसे ज्यादा मीट का प्रोडक्शन अपने होता है। ये कंट्री वाइज है, अगर मीट और मेरे हिसाब से जो मिल्क है सबसे ज्यादा इंडिया के अंदर है, फॉलोड बाय चाइना है, फिर बांग्लादेश है। ये ज्योग्राफिकल मैंने दी। नेक्स्ट, ये गोट का डिस्ट्रीब्यूशन जो अभी मैंने सबसे पहले कहा कि सबसे ज्यादा बकरियां किसके पास पाली जाती है, या तो जिनके पास जमीन नहीं है या जिनके पास 1-2 एकड़ जमीन है। ये 85% के करीब तो जो लैंडलेस स्मॉल या मार्जिनल फार्मर है वो ही बकरी पालन कर रहे हैं। अब पिछला जो सिनेरियो गया है 10 साल का, इसके अंदर एकदम चेंज हुआ है, जिसके अंदर बड़े बड़े जो फार्मर है जो कनाडा से या अमेरिका से रिटर्न हो रहे हैं वो अब बकरी पालन की तरफ जा रहे हैं। और पंजाब के अंदर बहुत सारे फार्मर हैं जो अभी कॉमर्शियल गोट फार्मिंग कर रहे हैं, और मेरे सारे के लगभग सारे टच के अंदर है, कंसल्टेंसी मेरे से ले रहे हैं कभी कोई प्रॉब्लम आ रही है। नेक्स्ट, उसके बाद जैसे मैंने आपको बता दिया कि पॉपुलेशन 148 मिलियन है, बायोडाइवर्सिटी अगर ब्रीड की बात करें 37 है इंडिया के अंदर, और मीट का प्रोडक्ट जो मिल्क का प्रोडक्शन है वो 5.88 मिलियन टन है, और मीट का है 1.17 मिलियन टन इंडिया के अंदर। नेक्स्ट, इकोनॉमिक्स का अगर पार्ट का है, अभी थोड़ा सा मैं आपको बता दूं, एग्रीकल्चर या वेजिटेबल, सबसे ज्यादा वेजिटेबल के अंदर है कौन कमाता है? यहाँ ज्यादातर मेरे को लग रहा है फार्मर फैमिली से भी बिलोंग करे हैं या एग्रीकल्चर स्टूडेंट है। वेजिटेबल के अंदर सबसे ज्यादा मिडल मैन कमा रहा है। आज की तारीख का मैं बता रहा हूँ, आज मार्केट के अंदर खीरा का जो रेट है वो 30 से 32 रुपए पर केजी चल रहा है, और फार्मर से 6 रुपए किलो जा रहा है। कंज्यूमर को 30 रुपए दिए में बिक रहा है। आप किसी भी रेडी से या मार्केट पे जाइए, 30 रुपए से कम आपको खीरा नहीं मिलेगा। फार्मर जब सुबह लेके जाता है तो उसको 6 रुपए किलो से ज्यादा नहीं मिल रहा है। ये पिछले 15 दिन का मेरा पर्सनल एक्सपीरियंस है। यानी सबसे ज्यादा किसने कमाया? कई गुना 1 घंटे के अंदर मिडल मैन ने कमाया। वो फार्मर 6 महीने मेहनत कर रहा है, उसको 6 रुपए किलो मिल रहा है। मिडल मैन जिसने 1 घंटे तक मेहनत की, वो उसके अंदर से 6 और 30 के बीच में कितना 24 रुपए का मार्जिन मिडल मैन खा गए। कोर्ट है, उसको देखिए आप, इसके अंदर है 74% मार्जिन है, वो गोट ब्रीडर का है, फार्मर का है। मिडल मैन इसमें भी कमाते हैं लेकिन बहुत कम, क्योंकि इसके अंदर 2-3 मिडल मैन आ जाता है, एक परचेजर आता है, और उसके बाद नेक्स्ट है जो वो स्लॉटर हाउस वाला कह सकते हो या बुचर खाने वाला, जो भी उसको काट के सेल करता है। नेक्स्ट, ये इकोनॉमिक्स है जो मैंने पहले ही बताया कि जस्ट डबल करती है। अगर 1 रुपया खर्च करते हो तो एवरेज 2 पॉइंट समथिंग उसका बीसी रेशियो आता है। नेक्स्ट, ये फैक्टर है जो क्या क्या उसकी प्रोडक्शन प्रोडक्टिविटी को अफेक्ट कर सकते हैं और कौन कौन से है, जैसे प्रोलिफिकेसी है, मिल्क यील्ड है, सर्वाइवल रेट है, फिनिशिंग रेट है, ड्रेसिंग परसेंटेज है, बोन टू मीट रेशियो है। ये आपकी बेसिक सी चीजें हैं। इसको बड़ा करो, कुछ सही नहीं है, शेयरिंग पे आता है, शेयरिंग पे आता है, नीचे
नीचे वो
जाइए, स्लाइड को बड़ी करिए, स्लाइड शो पे हाँ तो साइड वाला है ना। ड्रेसिंग परसेंटेज समझते हो? ड्रेसिंग परसेंटेज का मतलब है कि एक जिंदा जो लाइव वेट का 100 किलो का कोई अगर बकरा है तो उसके अंदर मीट कितनी मिलेगी, प्योर जो जिसको खाने के लिए यूज कर रहे हैं, यानी वो कितना परसेंट है। ठीक है, उसके बाद है बोन टू मीट रेशियो। इस चीज के अंदर थोड़ी सी बताने वाली है कि जो टोटल आया अपने को उसमें से बोन का कितना पार्ट है और मीट का कितना है। ठीक है, और फिनिशिंग वेट यानी लास्ट जिस टाइम पे आप सेल कर रहे हो उस टाइम पे उसका क्या रेट रहा है। सर्वाइवल रेट कितने परसेंट मोर्टेलिटी है और कितना परसेंट उसने सर्वाइव किया है। और मेन चीज आती है ब्रीडिंग, फीडिंग, हेल्थ केयर, हाउसिंग एंड मैनेजमेंट प्रैक्टिसेस है। ये बेसिक चीज है। नेक्स्ट, ये चैलेंज है, हालांकि इसको चैलेंज नहीं कहेंगे, चैलेंज होता है वही अपॉर्चुनिटी होती है। अगर उसके अंदर कुछ कमियां नहीं है तो आप क्या सुधार करोगे। इसलिए ये है थोड़ा सा पॉपुलेशन जो है वो ज्यादा है और प्योर जो ब्रीड है वो एक्चुअल के अंदर कम है। फार्म के बात करें तो उसके डाटा रिकॉर्डिंग बहुत कम है और इनडिस्क्रिमिनेट ब्रीडिंग यानी जो अच्छे नस्ल के बक है उनको ज्यादातर फार्मर सेल कर देते हैं ज्यादा पैसे के लालच में, और जो बिल्कुल ही कमजोर या जिसका रेट कम मिलता है उसको घर पे रख लेते हैं और वही है जो ब्रीडिंग बक के रूप में यूज होता है। ये कुछ कमियां हैं। ठीक है, और कैपेसिटी बिल्डिंग स्टेक होल्डर फार्मर को एप्रोप्रिएट या प्रॉपर ट्रेनिंग नहीं दी जा रही है। ज्यादातर जो अगर इस टाइम पे करें तो सोशल मीडिया है तो वो पता नहीं राई का पहाड़ बना के भी पेश कर देते हैं, क्या चीज है उस चीज से थोड़ा सा पता ही नहीं चल रहा है। ठीक है, उसके बाद प्रॉपर जो पैकेज एंड प्रैक्टिसेस है वो डेवलप करने की जरूरत है और ऑर्गेनाइज्ड मार्केट सबसे बड़ी चीज। हालांकि अब पंजाब के अंदर तो काफी ऑर्गेनाइज्ड मार्केट है जो जगह जगह लगते हैं बकरी का बाजार और लोग अपना बकरियों को वहां जाके सेल कर रहे हैं। ये गोट ब्रीड्स है जो 37 जो मैंने बताई, सबसे ज्यादा जो वेस्ट बंगाल वाला जो इलाका है ना नॉर्थ वेस्टर्न, सबसे ज्यादा गोट की पॉपुलेशन वहां पाई जाती है और दूसरे नंबर पे अपना जो पंजाब है, हरियाणा है, राजस्थान जो सेंट्रल जो पार्ट है उसके अंदर। नेक्स्ट, ये ब्रीड वाइज है दूध के लिए, जैसे ये मैंने बताया पंजाब की बीटल सबसे बेस्ट है, जमुना पारी है, जटराना है, सूरती है और बाकी सारे की सारी ड्यूल पर्पज है। और चांगथांगी और चेगू हिमाचल की जो दो ब्रीड है जो स्पेशल फर के लिए, फर प्रोडक्शन के लिए यूज होती है। नेक्स्ट, ये कुछ ब्रीड्स की ये है। नेक्स्ट, ये ब्रीड वाइज जो गोट है उनका दे रखा है। नेक्स्ट नेक्स्ट नेक्स्ट, ये एक्सोटिक ब्रीड्स है, वो पहले सारी की सारी इंडियन ब्रीड थी, कुछ इसमें बारी जो एक्सोटिक ब्रीड्स है वो भी यहाँ यूज करते हैं, जिसके अंदर सबसे बेस्ट जो बीच में सेंटर में देखिए बोअर, इंडिया के अंदर अगर क्रॉस ब्रीडिंग के लिए यूज की जा रही है वो है, उसका मीट ज्यादा रहता है। अपने वाली अगर मान के चलिए 50 किलो का वजन रहता है 1.5 साल के अंदर, उसका 1 क्विंटल 30 किलो तक पहुंच जाता है, यानी मीट का प्रोडक्शन ज्यादा है लेकिन मिल्क का बहुत ही नाम आता है। ये मैंने खुद ने भी रियल करवा के देखी है, 1 क्विंटल 30 किलो तक मेरे खुद के गया हुआ है। ये जो गोट की पोटेंशियल है कि कहाँ कहाँ कितना कितना है, उसके अंदर दूध की जैसे एवरेज 90 से 100 किलो रहता है, जबकि वो चाहे अगर अच्छा प्रोडक्शन आप लेना चाहते हो, मैनेजमेंट आपकी सही है, तो वो एक लैक्टेशन के अंदर यानी 400 किलो तक पहुंच जाती है। और अपने यहाँ मेरे खुद के गुरु अंगद देव वेटनरी यूनिवर्सिटी के हमारे जो फार्म पे है, हाईएस्ट जो बीटल का दूध प्रोडक्शन हुआ है वो 5 लीटर के करीब। एक दिन का मायना रखता है 5 लीटर। अच्छी अच्छी गाय या भैंस के भी नहीं होता, हमारे बकरियों का होता है। ठीक है, नेक्स्ट। ये कंजर्वेशन क्यों, तो उसके ऊपर पहले ही अपने चर्चा कर चुके हैं कि अगर सही हिसाब से जो अगर जेनेटिक वेरिएबिलिटी है वो खत्म हो जाएगी तो फिर कुछ नहीं बचेगा, इसलिए कंजर्वेशन भी जरूरी है। और नेक्स्ट, इसमें कंजर्वेशन के लिए अगर हम बात करें तो 2 सिस्टम होता है, इन सीटू और एक्स सीटू। इन सीटू जहां जिस फार्मर ब्लॉक के अंदर है वहीं पे ही उसका इंप्रूवमेंट करना होता है, और एक्स सीटू के अंदर आप उसका सीमेन है या जो भी है उसके द्वारा दूसरी जगह कर सकते हैं। गोट इंप्रूवमेंट प्रोग्राम भारत के अंदर सबसे बहुत पहले से चल रहा है, और सबसे पहले अगर बात करें तो आजादी से पहले भी अंग्रेजों के टाइम पे भी बाहरी नस्लें लाके अपनी इंडियन नस्ल को सुधारने का कुछ काम हुआ था, और 70-72 से भारत सरकार के द्वारा एग्री प्रोजेक्ट चल रहे हैं जो डिफरेंट ब्रीड पे काम कर रहे हैं। अब ब्रीडिंग अप्रोच क्या है, सिलेक्टिव ब्रीडिंग, और दूसरा है ग्रेडिंग अप और नॉन डिस्क्रिप्ट। सिलेक्टिव का जानते हो, कभी आपका क्वेश्चन आ सकता है कंपटीशन के अंदर, व्हाट इज द सिलेक्टिव ब्रीडिंग। सिलेक्टिव ब्रीडिंग का मतलब है जो अच्छी नस्ल है, जहां की ब्रीड है उसी में ही प्योर ब्रीड को सिलेक्ट करके आगे ब्रीडिंग के लिए काम में लेना, यानी सिलेक्शन करना। ठीक है, दूसरा होता है ग्रेडिंग अप, नॉन डिस्क्रिप्ट। उसके अंदर मान के चलिए आपके पास बिट्टल बकरी है, बहुत अच्छा है, उसका बक लोगे आप, और जिसके फार्मर के पास बिट्टल नहीं है, नॉन डिस्क्रिप्ट टाइप है, देसी टाइप है, उसके वहां उस बक को ब्रीडिंग करवाओगे तो उससे नेक्स्ट जो जनरेशन आएगी उसके अंदर 50% बिट्टल आएगा, नेक्स्ट आएगी 62 आएगा, नेक्स्ट आएगी, ऐसे करके उसको 5 से 6 पीढ़ियों के अंदर आप प्योर बिट्टल के अंदर कन्वर्ट कर दो। ठीक है, नेक्स्ट फीडिंग का मैंने पहले ही आपको बता दिया, ये ब्राउजिंग करती है, ग्रेजिंग नहीं करती है, नीचे गिरा हुआ नहीं खाती है। नमकीन इसको बहुत स्टूडेंटों को आता है ना आजकल, चटपटा नमकीन खाना ही, हमारे बकरियों को भी नमकीन और चटपटा खाना ही सबसे अच्छा लगता है। ठीक है, और उसके अंदर भी लेग्यूमिनस पाउडर है वो सबसे ज्यादा पसंद करती है, और न्यूट्रिएंट की जो कन्वर्जन एफिशिएंसी है फॉर मिल्क प्रोडक्शन के लिए वो 38% डेयरी गाय के अंदर, गाय भैंस में है ना, और नीचे गोट में देखिए 45%। उसके बाद ड्राई मैटर इनटेक, यानी शरीर भार का कितना वो बॉडी वेट के अनुसार कितना ड्राई पाउडर इनटेक करती है, तो गोट जो मीट गोट है 3 से 4% है, और डेयरी के लिए 5 से 7% है। ये क्वेश्चन आप लोगों के लगभग हर जगह आएगा, कभी कंपटीशन में बैठोगे तो। ठीक है, नेक्स्ट, ये जो फीडिंग का है जो मैंने बताया, एक एक्सटेंसिव सिस्टम पूरा का पूरा ग्रेजिंग कराना, दूसरा सेमी इंटेंसिव जिसके अंदर कुछ घंटे आप 4 से 6 घंटे चला के लाओगे और बाकी घर पे फीडिंग करते हैं, और एक होता है इंटेंसिव यानी कंप्लीटली स्टॉल फीडिंग। नेक्स्ट, अगर प्रोडक्शन परफॉर्मेंस की बात करें तो सबसे बेस्ट होता है सेमी इंटेंसिव, यानी कुछ घंटे उनको ग्रेजिंग करवाओ और बाकी घर पे स्टॉल फीडिंग करवाओ। ठीक है, नेक्स्ट, सेल्टर की है, तो सेल्टर के अंदर इनको एलिवेटेड जो मैंने पहले ही बताया है ना, सिलेक्टेड हाउस है वो सबसे ज्यादा उनको पसंद है, यानी अच्छी जगह पे जाके, ऊपर की जगह पे जाके बैठना पसंद करती है। नेक्स्ट, ये डिफरेंट टाइप ऑफ हाउस है, ये तो एक नेक्स्ट, स्पेस रिक्वायरमेंट है, अलग अलग बच्चे को कितना चाहिए, बड़ी को कितना चाहिए। नेक्स्ट, ये डिफरेंट हाउस है जो बकरियों के लिए, ये मेरे पर्सनल फोटो खींचे हुए हैं इंडिया में डिफरेंट जगह पे जाके। नेक्स्ट, ये हेल्थ मैनेजमेंट है, कौन कौन सी इनको बीमारी आती है, किस किस का वैक्सीनेशन करना चाहिए, डिवर्मिंग करना चाहिए। ठीक है, सबसे ज्यादा पीपीआर, ईटी, ये इनकी सबसे ज्यादा खतरनाक बीमारी है। नेक्स्ट, जो इनकी प्रोडक्शन और प्रोडक्टिविटी को डाउन कर देती है। फील्ड लेवल पे आज भी 27% के करीब इनकी मोर्टेलिटी है और अडल्ट की 10% के करीब। ठीक है। नेक्स्ट, ये जो वैक्सीनेशन का शेड्यूल है कि कौन कौन सी वैक्सीन कब लगवानी है। नेक्स्ट, ये जो डिवर्मिंग वगैरह करनी है, डिवर्मिंग का सबसे बेस्ट है प्री एंड आफ्टर मानसून। नेक्स्ट, ये स्ट्रेंथ जो मैंने बताया बात के अंदर बहुत ज्यादा है। नेक्स्ट, वीकनेस क्या है वो भी अपने चैलेंज के अंदर लगभग डिस्कशन कर चुके हैं। नेक्स्ट, अपॉर्चुनिटी क्या है, तो जो चैलेंज है वही अपॉर्चुनिटी भी है आपके लिए। आपके पास बहुत बड़ा सेक्टर है काम करने का, जिस पे चाहे उस पे आप काम कर सकते हैं। लार्ज जेनेटिक वेरिएबिलिटी आपको मिलेगी। अगर आज आप कोई ब्रीड भी डेवलप करना चाहते हो तो भी आप आराम से कर सकते हो। अभी भी बहुत सारी जो ब्रीड है उसके ऊपर टच भी नहीं हुई। नेक्स्ट, ये है सीड क्या है। सबसे पहले जो मैंने बताया जेनेटिक डायल्यूशन हो गया। पंजाब के अंदर बीटल थी, लोग सिरोही लेके आ गए, कुछ लोग जमुनापारी ले आए, कुछ लोग बारबरी ले आए। बारबरी वाला पूरा का पूरा बेल्ट है उसने सिरोही ने खराब कर दिया। मतलब जहां दूसरे ने किसी ने बता दिया राजस्थान वाली बकरी बहुत अच्छी है, पंजाब वाले भूल गए कि हमारी बीटल सबसे अच्छी है, उसको भी लेके आके और क्रॉसिंग होगी वो सबसे खतरनाक। ठीक है। और जो अनकंट्रोल ब्रीडिंग है वो मैंने बता ही दिया आपको। नेक्स्ट, ग्रेजिंग का जो सोर्सेज है वो डे बाय डे है वो इंक्रीज हो रहा है। ये स्पेसिफिक रिकमेंडेशंस है। ब्रीडिंग गोट ब्रीडिंग सोसाइटी बननी चाहिए। अपग्रेडेशन ऑफ नॉन डिस्क्रीट जो गोट है वो थ्रू प्योर ब्रीड से अपग्रेडेशन होना चाहिए। और एआई टेक्नोलॉजी का यूज होना चाहिए, यानी डायरेक्ट बक को ब्रीडिंग करवाने की बजाय एआई अगर करें तो ज्यादा बढ़िया। ठीक है। फीड रिसोर्स डेवलपमेंट करना, गोट हेल्थ पे देना, और सबसे ज्यादा है ऑर्गेनाइज मार्केटिंग सिस्टम और वेट वेस्ट के ऊपर गोट का मार्केटिंग होनी चाहिए। एक्सटेंशन के लिए ट्रेनिंग, एक्सटेंशन फंक्शनरीज के लिए ट्रेनिंग। कुल मिला के कहने का मतलब भाव ये है, बकरी पालन के अंदर बहुत ज्यादा स्कोप है। हर साल एफडी पकाती है, घाटा होने का कोई भी चांसेस नहीं है। लेकिन अगर आप ये चाहो कि एक साल में करोड़पति बना देगी, असंभव। ठीक है। नेक्स्ट, अब ये कुछ मेरे लिए फोटोएं हैं। पूरे इंडिया के अंदर डिफरेंट साइज के फ्लॉक है, किस टाइप में हाउसिंग में रख रहे हैं, कैसे रह रहे हैं, लगभग शायद मैं भी कहीं लिख जाऊंगा आपको। हाँ, नेक्स्ट 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 नेक्स्ट। थैंक यू।
Thank you, sir, for your insightful presentation on prospects of horse farming. We all are truly grateful to you for sharing your valuable knowledge with all of us. Now, I would like to welcome Dr. Amita Mahajan, Actually Agriculture Department, and Dr. Manoj Pali, Dean, USAS, for being welcome and a token of appreciation to our judges. Now I would like to request Dr. Magardeep Singh sir to come up on the stage for welcome. I would like to request Dr. Dr. Pavandeep Singh sir to come up on the stage for token of appreciation.
पता नहीं इतने भैया थी उन्होंने देती मैडम हाँ जी मैं कॉपी कर लिया ठीक है हाँ जी कोई सी ਇਹਦੇ ਵਿੱਚੋਂ आरबी मैडम दस्यो मैनू केड़ी है हाँ तेरा फिर पेज ये दिखा रहा है कोई किसी फोल्डर में तो नहीं रखी हुई है किधर गया
Thank you, everyone. Now let us have the pleasure of listening to our next esteemed guest, Dr. Pavandeep Singh sir, former professor and head of department, Chief Scientist, Environment, Biotechnology, Microbial Biochemistry, CSIR, Council of Scientific and Industrial Research, Chandigarh. He was awarded with a prize by Director General of CSIR for 2 of his publications that were listed in top 17 publications of CSIR. We all are extremely delightful to have the one whose name is listed in top 2% of global scientists. And now I am honored to invite him to deliver his thoughts on importance of microbes in agriculture. So ladies and gentlemen, please put your hands together for Dr. Pavandeep Singh sir.
एक कमेंट यार लापे आर के नाम तो आगे बोल रहा है कोई फोल्डर ही है नहीं हेलो आरी आर के नाम
खोल के दिखा दो पता लग आरबी ना जेड फोल्डर से
रोल ऑफ इन कंट्रोल इसे कॉपी कर लो ਇਹਦੇ ਵਿੱਚ ਕਾਪੀ ਕਰਿਓ ਠੀਕ ਹੈ ਨਾ ਸਰ ਅਲੱਗ ਫੋਲਡਰ ਦੇ ਵਿੱਚ ਬਣਾ ਲਓ ਅਲੱਗ ਫੋਲਡਰ ਦੇ ਵਿੱਚ ਕਾਪੀ ਕਰ ਲਿਓ ਪੈਨ हाँ जी, ना लगा। हाँ जी, कलर नहीं सही है तो ਇਹਨਾਂ ਦੀ ਬੈਕਗ੍ਰਾਉਂਡ ਵੀ ਸਹੀ ਨਜ਼ਰ ਆ ਰਹੀ ਬੈਕਗ੍ਰਾਉਂਡ ਸਰ ਤੁਹਾਡੀ ਬੈਕਗ੍ਰਾਉਂਡ ਜ਼ਿਆਦਾ ਡਾਰਕ ਕੀਤੀ ਹੋਈ ਹੈ ਪ੍ਰੈਜੈਂਟੇਸ਼ਨ ਦੀ
It's some problem in the PPT, but anyway, I'll talk to you. Thank you very much for the introduction. Such a nice introduction. I'm very grateful to Dr. Amita Mahajan and her team for inviting me to this international conference. Today I'll speak to you of the role of microbes in agriculture. Because I'm a man of microbiology, so I'll talk more about bugs.
लाइट ऑफ
Next one. Okay. Anyway, microbes play an important role in agriculture, soil health, plant nutrition, and disease suppression. Microbes help, as we all know, in nitrogen fixation, nutrient cycling, and pathogen suppression. Microbes reduce the use of chemicals like pesticides, herbicides, and insecticides in agriculture. They promote soil fertility and help in plant growth. Microbes can shape agricultural ecosystems. There are three major roles of microbes in agriculture. The first one is biocontrol agent. The second one is biofertilizers. And the third one is production of biodiesel. The other roles of microbes in agriculture are bioremediation, nutrient management, plant growth regulators, biocomposting, bioleaching, and natural formula. Let us first go to biocontrol agents. In biocontrol, biopesticides are very, very important. They do not have any chemicals like in herbicides and pesticides. They are all live bacteria. They produce antibiotics and siderophores. They kill and control pests. Examples are Bacillus thuringiensis, BT strain. We call it BT, which produces toxins lethal to insects, pests, and fungus species like Beauveria. These pesticides offer targeted pest control, and they do not kill non-target pests and are beneficial to the environment. Next, we come to biofertilizers. At least you can see the bacteria. Biofertilizers are living microbes. They are live. They are very much living, and they are very smart. The biofertilizers apply to the seeds, saplings, and to the soil. Before sowing the seeds, they are put in biofertilizer. There is a coating of biofertilizer on the seed, on the saplings, and by spraying in the soil. They enhance plant growth by increasing the nutrients. These microbes fight pathogens by producing antibiotics and siderophores. Examples are Rhizobium, Acetobacter, Azospirillum, Bacillus, and Penicillium. They all promote plant growth. Coming to biodiesel, this is very, very important economically. It is produced from a variety of feedstocks: soybean oil, canola oil, palm oil, waste vegetable oil, and animal waste. Microbes like algae, yeast, and fungi have a very high lipid content, which can be extracted and converted into biodiesel fuel. Microbes can be grown in a big fermenter, 100 liter, 200 liter, 1000 liter. The microbes can be grown in the fermenter, and they can produce biodiesel in large quantity. Next one. These are some of the microbial fertilizers, biofertilizers, like Acetobacter, Frankia, Rhizobium, and Methylobacterium. They produce phytohormones, siderophores, nutrient enrichment, and stress resistance. You can see that they solubilize phosphate, and the mycorrhiza is there in the root nebule, and they also fix nitrogen. Next one. Now coming to bio-leaching, this is very important. This is leaching of metals and metal ions into the system. Bio-leaching is a process of mining between microbes and minerals. Microbes extract valuable metal ions from ores. Examples are Pseudomonas, Bacillus, Thiobacillus, yeast, Aspergillus, and Penicillium. This is a very good metal which we developed on metal recovery using microbes. And this was published in a very good journal known as EDRC. And this model is used by many people around the world. You have a metal in soil, you put biosurfactant inside, you get a complex, metal biosurfactant complex, then you flush it with water, and you get metal recovery. This is very important for precious metals like gold and tungsten. Coming to my favorite topic, that is bioremediation. Bioremediation is a process where microbes like bacteria, fungi, yeast are used to degrade pollutants in the soil and water. You can see the rods, they are all Bacillus species. Microbes degrade pollutants in water and soil. Now, metals cannot be degraded. You cannot degrade iron. You cannot degrade gold. But you can only transform the metals into less toxic forms. Supposing some metal is toxic, you can transform the metal using microbes
into less toxic form. This is what microbes do. You know, especially in Punjab, they spray a lot of pesticides, herbicides, insecticides to the farm, and this persists in the environment. They cannot be degraded, and after some time, it seeps down to the drinking water, to the groundwater. But our friends, the microbes, bacteria, fungi, they silently, quietly degrade toxic compounds like pesticides, herbicides, oil sludge, etc., and make the environment green, make it clean. Next one. This is a cartoon from Friends in Biotechnology. These are bacteria which are eating pollutants like dyes, oil sludge, petroleum hydrocarbons, and pesticides, herbicides, and insecticides. They love it. They eat it very easily. Like sugar. Like we like sugar, we like honey, we like pollutants, and they clean our environment. This is the toxic compounds in crude oil sludge. You know, there are a lot of wineries in Assam and in Bombay High and Gujarat. There are a lot of oil refineries. They produce a lot of oil sludge which contaminate the environment. Now our friends again can degrade all these toxic compounds. Next one. Next is nutrient management. Microbes optimize soil conditions to improve nutrients. They decompose organic matter, they fix nitrogen, as it is shown here, and they also solubilize phosphate and they solubilize potassium and sodium too. So they are very good for the nutrient management. And the species like Aspergillus and Rhizobium are very, very important. Next. This is a list of some of the beneficial microbes which help in agriculture, like Bacillus, Pseudomonas, and Enterobacter, Streptomyces. They all help in agriculture. They are associated with crops like wheat, pepper, coffee, wheat, soybean, oil palm, chili, tomato, etc. Let us see the case of chili. There is a bacteria known as Pseudomonas. This makes siderophores which produce iron. And iron is a very important metal for the growth of chili. So they provide more iron for chili growth and it helps bumper crop of chili. Next. These are the factors affecting microbial communities in agriculture. The first one, as we all know, is crop rotation. The second one is fertilization. And the third one is tillage. Crop rotation is very, very interesting. You have roots like carrot, you know, you have radish, you have turnip, then you have the potato family, which comprises of potato and tomato. Then you go to legumes. You have peas, French beans, etc. Then you come to the Brassica family, which is cabbage and cauliflower, radish, and so on. So if you do crop rotation, you better bumper crop. And then tillage is very important. You know, even at home, in your pots, you do some tillage, right? So that more oxygen goes inside. So bacteria can help you if you provide more oxygen to them. And you have to irrigate also. Irrigation पे आपको पानी डालना, पानी डालना बहुत important. You have to irrigate the farm very well. This is a little bit of microbiome. This is a collection of bacteria, a collection of fungi. The microbiome is studied in 3 ways. First one is culture dependent method, in which you do plating of microbes. So you grow the microbes in a plate, and this is underestimation of biodiversity. You can only know a fraction of the biodiversity. So you grow on the plate, those colonies on the plate. Come to this one, microgram. Microgram, next one. Yes, yes. Microgram. Yes. And there is another method known as culture independent method, in which there is overestimation of microbes. You come to know about trillions and trillions of microbes in the soil. And the first one is microbiome composition. What is the composition of the microbiome? And you do amplicon sequencing. I cannot tell you everything. I'm just giving you introduction. Okay? And then there is a software for analysis and results. The second method is microbiome function. The function of the microbiome. So here you do sequencing of all the genomes, and then there is a software for analysis and results. Next one. Now there are a lot of metals in the soil, metal ions like calcium, magnesium, iron, sodium, copper, zinc, etc. These are all in the ion form. The microbes mobilize the ions for the plants to take up. So metals are mobilized or transformed by microbes to less toxic forms, and they are provided to the plant for the growth. Next one. What can we do for the soil biodiversity loss? How can you save the soil microbes? You must have living soil. You know, it should be full of earthworms. It should be full of microbes, bacteria, and yeast, and also animals and insects. Let the animals go there, rabbits and cows and whatever is there. Let them go there. Insects, so that the soil is not dead. It is a living soil. It is a live soil. There you can do better agriculture than a dead soil. Right? So these are the importance of bacteria and fungi in agriculture. So the take-home message is conserve living soil for agricultural biodiversity. You should also give a lot of money, good irrigation to have a good agriculture. And we should conserve the plants, animals, bacteria, virus, insects, fungi, protozoa, algae, and cyanobacteria. So that they help us in growing good crops. If you have any question, you can ask. If you have any questions, you can ask. Okay, then good luck to you.
Thank you, sir, for your outstanding presentation at our event. Your expertise on importance of microbes in agriculture captivated our audience and left a lasting impression on all of us. It gives me immense pleasure to introduce our next distinguished keynote speaker, Professor Dr. M.C. Sidhu, sir from Department of Botany, Punjab University, Chandigarh. His substantial contribution in field of angiosperm taxonomy, medicinal plants, and plant breeding left an indelible mark on countless of individuals. He has teaching and research experience of about 21 years. We are truly honored to have an opportunity to hear the enlightening words today. So without any further ado, let's welcome Professor Dr. M.C. Sidhu with a big round of applause.
How much time is allotted? 15 to 20 minutes, right? I think actually it is the lunch time, basically, right? So I am standing between you and your lunch, right? So I hope that all these students those are sitting, they are from the agriculture fraternity. Yes, please, students, you are from agriculture background. So I believe that you know so many things about agriculture. I am from botany side, right? My department is botany, Punjab University, right? So let's
let us talk about plant resources that can help in agriculture, you can say toward food and healthcare system, right? Because when we talk about the agriculture, in agriculture there is a plant breeding, right? And plant breeding is directly related to the botany. And if you know the basics of the botany, then you can know about the plant, you can identify the species, then you can do the plant breeding, right? Normally we say that let us cross two plants with each other so that you can have a better progeny in the apple generation. But until or unless you are aware about the plants or the diversity or plant diversity or crop diversity or species diversity, it is not possible, right? So if any of the students, what is the current population of India? Any of the students? One, not it is 143 population of India, 143 crore. And right now we are at number 1 or 2? We are number 1 and China is at number 2. So why I am asking this question? Because when we are number 1 in terms of human population, so it creates so many concerns. Next slide, please. So if we look at the human population, right? So it is 143 point something in case of India and China is next to us, right? So previously it was thought that the population will be 8 billion in the year 2027, but we have reached 8 billion even in 2023. So it shows that we are moving very fast towards the increase in human population. And if the population is increasing, what are the requirements to build the ever human population? We need more houses, more food, more school, more colleges, more industries, right, etc., etc. And for that we need space. And if population is more, so space is limited, right? We cannot increase the space. Then what to do? Next, please. So there are some basic requirements of the human being. You can say food, fuel, timber, etc. You can say medicines are quite important, vegetables, spices, shelter, etc., etc. Right? So these are some of the basic requirements which plants only can, you can say, plant can help us to achieve all these requirements. Next, please. So if we have to manage the food requirement of the increasing population, how we can manage? Number 1, increasing the area under cultivation, we can increase the area under agricultural crops. Second, promotion of the soil properties, because soil properties are very important in terms of yield of agricultural crops. So we have to improve the composition of the soil. We can use genetically modified crops, benefit of high yielding varieties, or we can reduce the wastage of the crop. Next, please. So when we say let us increase the area under cultivation, is it really possible? So look at the literature. What the literature says that every day, every moment, every second, we are losing arable land. Arable land means the land which we are using for agriculture, right? So when we are making so many houses, residential complexes, new roads, widening of roads, etc., etc., new industries, they are coming up. So we are using the agriculture land. So to my mind, under these circumstances, it is very difficult to increase the area under agriculture. Next, please. But the soil properties can be improved because whenever there is a crop in the agriculture field, so microbes they grow on the plants, ultimately they you can say mix in the soil and they change the chemical properties of the soil. And sometimes these chemical properties they are good and they are ultimately helping us to increase the yield. So previously, Professor Swanji Singh he has also talked about the soil microbes that how they can help us to increase the agriculture field. So when we talk about genetically modified crops, so do you have any idea that what is the status of genetically modified crops in India? Any student? You are from agriculture background. So what is the status of GM crops? Anybody? Do we grow genetically modified crops in India? How many crops and which one? If your department, if you are BSc agriculture student, you must know that what is the status of the GM crop. genetically modified crops, so in which the genomes have been modified through genetic engineering. That's it. So if we look at the genetically modified crops across the world, that USA is number one, Brazil number two, then Argentina, and India is at number 4 in terms of the area under cultivation. But next slide. Interestingly, in India we have only one crop, which is cotton. So in India, cotton is the only crop which we cultivate at the commercial level. So it was released in 2002 by the Maharashtra hybrid company. And initially it was released for 5-6 states, but now I think it is cultivated all over India. So there are more crops like you can say it was released, but later on it was withdrawn. Even in the last year, you can say GM mustard was released, but due to obesity reason, it was again you can say withdrawn. The matter is subject matter. Next please. So the area under in the 2003 area was you can say even less than 1 lakh hectare. But in the year 2018-19, the area under British cotton cultivation has been increased extensively. So, if we look at these three terms, invasive species, then herbicide resistant crops and herbicide resistant weeds. So, can any student make a difference between herbicide resistant crops and herbicide resistant weeds? What do we mean by this? When we say herbicide resistant weed, what does it mean? That any weed on which herbicide is not effective. It means that weed has become something like a super weed, right? So when it is very difficult or weed cannot be food, it means it will compete with the agriculture, our main crop. So this is the problem of the you can say herbicide resistant weeds. Similarly, if you talk about the herbicide resistant crops, in this case, the genetic engineering is used, the crop is made herbicide resistant. In this case, our crop will not be affected by the herbicide, but the rest of all the species of the weeds which are growing, they will be affected, right? So it is not good for the environment. And in terms of invasive species, so these species they act as weed and they compete with the main crop for the food. You can say food, life, and space. That's good. Next, please. I am also in a hurry. Please wait. Right. So we can hybridize the crop plants, right? So you are agriculture student. I expect these things from you, right? Because you are in a hurry, right? But these things, I think, a little bit of important for you. So who made the first hybrid? Do you know anything? Because in agriculture, we normally say, let us hybridize the plants, hybridize the crops. So you know who made the first hybrid? It is Thomas Fairchild who made the first hybrid between these two species. Yes, please. So in case of wheat, according to Valley law, there are 14 species of wheat, but normally 4 wheat species they are cultivated. So what is the botanical name of the wheat which we are normally consuming? Any students? It become astragalus. It is a hexaploid wheat, right? So hexaploid wheat, then diploid wheat and tetraploid wheat all are cultivated, but more than 90% it is a hexaploid wheat. Rest of the species very less in cultivation. While addressing upon these 14 species, that these species can be used for hybridization programs. Next, please. Similarly, this slide shows a diploid, tetraploid, and hexaploid wheat. Next, please. Next, please. So, if we look at that, this is for 2 species of the, you can say, Triticum gyrandus and Aegilops. They have produced the tetraploid wheat, right? It means Aegilops is a different species, then Triticum is a different species. These are both are member of family Poaceae, if you know about the family, right? So this is how this tetraploid wheat has been found. Next, please. And from this tetraploid wheat, during the course of evolution, and around 10,000 years back, this hexaploid Triticum astragalus has been produced. Next, please. Now there is one wheat known as Kamut wheat. So this wheat was, you can say, in the mid 20th century, it was cultivated. But later on, with the release of the high yielding variety, this wheat we are not right now, we are not cultivating. Next, please. Now, if we look at the rice, in case of rice, there are 24 species, but only two species, that is Rhiza saiva and Rhiza piccorum. These are two widely cultivated. Rest of the species they are not cultivated throughout the world. Next, please. In case of cotton, there are 50 species. In case of cotton, 45 species they are deployed and 5 species which are tetraphytic. And out of these 50 species, 2 species of diploid they are cultivated, 2 species tetraphyte they are cultivated. The rest of the species they are available with us, and we can take different accreditation programs so that we can have a better variety in the future. It means there is a huge number of plant genetic resources which we can use for our agriculture program. Next, please. Similarly, this slide shows that for this tetraploid present day cotton has been formed around 7.5 million years ago. There was a common ancestor of the cotton, right? And around 1.5 million years ago, two diploid species they combined together and they have given rise to these 5 tetraploid species. Next, please. In case of potato, probably the potato which we eat is a autotetraploid potato. But a potato otherwise exists in diploid, tetraploid and hexaploid. When quality level variation is there, we can make different process so that we can have a better cultivars in case of potato as well. Next, please. In case of Arachis is peanut. Arachis like also when we say peanut, we will always. The genus Arachis, there are 80 species, but out of 20, only 2 are cultivated. One which is we call as a peanut, Arachis lycosia. Another one is Arachis labrata, which is the powder. The rest of the species, it means the breeder or the researcher, they have not many sources in the genus Arachis, which they can use for the breeding purposes. Next, please. Yes, I need water that. So, the case is not so simple that we have a genetic resources and we can make a cross with each other. Right? Not, it's not always possible. Like in the one case which was happened in 1927, 1928, the searcher was G.D. Karpichenko. He was a Russian geneticist. What he tried? He made a cross with radish and cabbage. In case of radish, radish is the underground part, if we are interested in, right? And in case of cabbage, the above ground part is useful. Why he made this cross? Radish and cabbage. He wanted that the underground portion of the radish and the above ground portion of the cabbage will combine together. We can have both radish and cabbage on a single plant. But what happened? Opposite. Root of the cabbage and leaves of the radish. It means nature do not allow us to do each and everything which we want. There are restrictions put by the nature. Right? Next, please. So next, how we can manage the food requirement simply. We should avoid the wastage. We can use alternate or traditional food. Like to this slide I have shown. Next, please. So food, why we are facing a difficulty in terms of food? Because that we are going far away from the traditional foods. Right? So what are the reason? A loss of knowledge about which local plants are edible. आज की डेट में मुझे कम पता है क्योंकि हमारा फील्ड पे जो है वो कम हो गया है तो वो एक रीजन है सुपरमार्केट आ गई सुपरमार्केट में क्या है घर बैठे मोबाइल पे जो भी वेजिटेबल चाहिए मंगवा लेते हैं नेचर में सराउंडिंग में जो भी फूड मटेरियल अवेलेबल है उससे हमारी इन्फ्लेशन है वो कम हो गया है नेक्स्ट प्लीज 2023 को ईयर को सेलिब्रेट किया था एज ए इंटरनेशनल ईयर ऑफ द नट्स द पर्पस वाज ओनली टू डाइवर्सिफाई द फूड राइट when food items they get diversified automatically there will be less pressure on the main food crops next please plant diversity if you talk about plant diversity towards the human health next please so there are you can say 312 plant species which we have reported from the literature which are antidiuretic, right? Normally, diabetes is a very common disease in every family. You can say hereditary, you can listen to the people that are talking about that diabetes is a very serious. So in plants, right, people they are using some plant ABCD for curing their diabetes, right? Even herbal medicines, people they are using. So it means plant resources, there are more than 300 plants which we can use as antiseptic. No doubt we need more and more scientific studies to make their use. Next, please. And similar such studies have been made in district Mandi, where the species of antigen has been noted. Next, please. Similarly, in case of district Una, Himachal Pradesh, information was gathered from more than 500 respondents, and they have shared 84 species which are anti-diabetic in nature. Next, please. So during in our study on the phytochemicals in case of plants, so we have come across you can say anti-HIV compounds which are present in these plants. These plants like Euphorbia meta, these are growing in you can say wild. Some of them they are herb and Columbellia is a tree, right? Terminalia pendula and Verdendia, they are also tree species, right? So we have just by doing their ECMS analysis, we have identified these compounds. But the major job is to isolate them and really see their activity whether in real sense they are anti-HIV or not. The job is pending, right? Next please. Similarly, in case of cancer, so we have you can say reviewed the plant species which we can use as an anti-cancer around you can say 5-6 years back. There are more than 1,000 plant species which are having anti-cancerous properties. But again, as I said, that only knowing the cancerous property in a particular plant or species is not important until or unless we isolate them and see their effectiveness in the human being. So malaria, you know that you name it has been accepted from the plant that cut it short. Next, please. Similarly, this lady, the name is very interesting. Name is you name of the lady. She's a lady and she has been shared a Nobel Prize, you can say, in the year 2015. And the research which they have done, Artemisia annua is a plant, and they have accepted artemisinin, which is a compound, antimalarial compound, right? So the interesting thing is that when they made a extraction through different solvents, the artemisinin was not effective at all at integrating. But when they extracted the same compound through ether, right? Ether, depending on the boiling point, the boiling point was low. When the same compound was extracted at less temperature, there was a 100% anti-malarial activity of this compound. Next, please. Now, the year 2026, after you can say 2 years, the year 2026 has been declared as the International Year of the Women Farmers. So, USA has proposed this, you can say, activity at the United Nations General Assembly. They have accepted it that year 2026 to be celebrated as the International Year of Women Farmers. So why the focus is on women farmers and what that agencies or different societies at the national or international level, NGO and all other societies, what they will do, what they will plan, what type of programs will be there. Number one, to increase the awareness about crucial role of women farmers in agriculture food system around the world. Then their contribution to food security, nutrition, and poverty eradication. The adoption of effective policies and actions against the barrier and challenges that women farmers face across every food system, and so on. Next, please. I think that's all for today. So there are some. Yes, so there are some, I think, pictures of the plants just for your consumption before plants, right? So any question if you have, then any questions related to this talk or any other, so you are welcome.
Thank you to you for the time and effort you dedicated in sharing your valuable knowledge. Once again, thank you for being an integral part of our event. Now, I would like to request each one of you to please stand up for national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जगति तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Now, I would like to request each one of you to come forward for a group photo. So this is the lunch time now. We will resume here at 2:30. Please be punctual and kindly promise please return at the given time, that is 2:30. Enjoy your lunch. Thank you.
Hello. Hello. Dr.
Hello.
Anand, you are the host.
Hello. हाँ जी डॉक्टर साहब।
हेलो डॉक्टर सर। हाँ जी।
हाँ जी सर।
हाँ जी, आपने प्रेजेंटेशन भेजी है?
हाँ जी मैं ਤੁਹਾਡੇ ईमेल ਤੇ ਭੇਜੀ।
क्वेश्चन वो बोल रहे थे एनी क्वेश्चन, आप बोल रहे थे हाँ, तो उसके बाद उन्होंने बीच में बोला ना, आपको हमें आवाज ही नहीं आई, लेकिन यहाँ पे फोन पे आ रही थी ना आवाज उनकी। मैंने मैसेज कर दिया। There is an internet, this power to internet connection with doctors. As it is terrible operation, उन्होंने कुछ leak कर दिया। उन्होंने फिनिश कर दिया था, लेकिन वो और आपसे लंबा चौड़ा लेक्चर एक्सपेक्ट कर रहे थे कि हाँ भाई वो यही था ना सर, वो लंबा चौड़ा वो एप्रिसिएशन वो कुछ कर रहे थे। हाँ वही वो, और वो वर्ड्स बुक कर रहे थे, आपने एक लाइन में खत्म कर दिया ना, तो वो आपकी मूवी हो गई।
मैम वहाँ तो दिन आए हो तो आपने जैसे बनाया हुआ आज। अच्छा आ जाओ मैम, आ जाओ, स्टार्ट हो गया। ठीक है, ओके मैम, थैंक यू।
मैक्सिमम 5 मिनट्स है आपके पास, मिनिमम कितने हैं सर? फोन पे पैड रखने के लिए हाँ करा दे
Dr. Nisala, my name is Abdul Arman. My topic is understanding climate change, its causes and impacts and mitigation strategies. So, first is what is climate change? So, climate change refers to the significant change in global temperature and weather pattern over time. बात करते हैं causes of climate change. तो जो ग्रीन हाउस गैसेस है इसमें एक प्रॉपर्टी होती है कि ये हीट्स को ऑब्जर्व करते हैं रेडिएशन को जो सनलाइट से आती है और फिर ये टेंपरेचर रीच करता है तो इनका कंसंट्रेशन हमारे एटमॉस्फेयर से ज्यादा हो जाता है तो जो मेजर ग्रीन हाउस गैसेस है वो है कार्बन डाइऑक्साइड मीथेन नाइट्रस ऑक्साइड एंड फ्लोरोफ्लोरोकार्बन तो डिफरेंट सोर्सेस हैं जो प्रोड्यूस करते हैं ह्यूमन एक्टिविटीज है जो प्रोड्यूस करती हैं ग्रीन हाउस गैसेस जैसे डिफॉरेस्टेशन कटिंग ऑफ ट्रीज जैसा कि हम जानते हैं जो ट्रीज होती है वो कार्बन डाइऑक्साइड ऑब्जर्व करती है और ऑक्सीजन देती है जब ट्रीज को कट कर देते हैं तो कार्बन डाइऑक्साइड का एमिशंस हमारे एटमॉस्फेयर में बढ़ जाता है और लीडिंग टू टेंपरेचर इंक्रीज हो जाता है देन उसके बाद आ जाती है एग्रीकल्चर एक्टिविटीज तो जो एग्रीकल्चर एक्टिविटीज है स्पेशली इन लाइमस्टोन मेथेन गैस हमारी प्रोड्यूस होती है उसमें लाइमस्टोन में जो मेथेन गैस होती है उसकी कैपेसिटी हीट ऑब्जर्व करने की कार्बन डाइऑक्साइड से कंपेयर में वो बहुत ज्यादा होती है और ये भी एक मेजर कॉज करती है क्लाइमेट चेंज का सो नेक्स्ट स्लाइड जैसा कि ये दिखाया गया है वो है डिफोरेस्टेशन हो गया और एग्रीकल्चर एक्टिविटीज हो गई और ये increasing population increasing population also responsible for producing high amount of carbon dioxide. इंडस्ट्रियल प्रोसेसेस, तो इंडस्ट्रीज में भी डिफरेंट प्रोसेसिंग इंडस्ट्री में हम बहुत सारी मशीनरीज का यूज करते हैं। जैसे कुछ मशीनरीज हो गई रेफ्रिजरेंट हो गया, एयर कंडीशनर्स हो गया, ये फ्लोरोकार्बन प्रोड्यूस करती हैं जो कि एक ग्रीन हाउस गैस है, वो भी क्लाइमेट चेंज कॉज करती है। माइनिंग एक्टिविटीज होगी, माइनिंग एक्टिविटीज में हम मिनरल्स को एक्सट्रैक्ट करने के लिए हाइड्रो मशीनरीज का यूज करते हैं, इसमें फॉसिल फ्यूल्स को बर्न करते हैं और वो कार्बन डाइऑक्साइड प्रोड्यूस करती है बहुत अमाउंट में, जो कि एक मेजर कॉज है क्लाइमेट चेंज। तो ये इंडस्ट्रीज एक्टिविटीज में जो इमिशंस होते हैं, इम्प्रॉपर वेस्ट मैनेजमेंट, जो हम वेस्ट मैनेजमेंट नेट क्लीन करते हैं उसमें भी मीथेन प्रोड्यूस होती है, और व्हीकल्स यूज करते हैं इसमें भी कार्बन डाइऑक्साइड प्रोड्यूस होती है, फ्यूल्स को बर्न करते हैं अगर। So impact of climate change on agriculture. So rising temperature, loss of biodiversity, increasing migration, reduction crop feed, land degradation, food insecurity. Next please. Impact on women, health risk, water scarcity, economic cost. So control measures of climate change. There are different control measures. First is reducing emissions. So जो emissions हो रहे हैं उनको reduce करना चाहिए, like renewable energy sources का यूज करना चाहिए, solar wind हो गया, hydro electric हो गया, ये यूज करने चाहिए हमें, इनसे emissions नहीं होते हैं जो कार्बन डाइऑक्साइड और कार्बन सिक्वेस्ट्रेशन, हमें एफोरेस्टेशन करनी चाहिए, रिफोरेस्टेशन करनी चाहिए। एफोरेस्टेशन में हम ट्रीज प्लांट करते हैं, रिफोरेस्टेशन में जिस एरियाज पे ट्रीज कट कर दी जाती है फिर से ट्रीज प्लांट करते हैं, वहां पे कार्बन डाइऑक्साइड और ऑक्सीजन का बैलेंस बना रहता है। सस्टेनेबल एग्रीकल्चर, हमें सस्टेनेबल एग्रीकल्चर करने से प्रैक्टिसेस अपनानी चाहिए जिससे मीथेन और नाइट्रस ऑक्साइड के एमिशंस को रिड्यूस किया जा सके, और ये प्रैक्टिसेस अपनाते हैं इसमें फार्मिंग, प्रिसीजन फार्मिंग हो गया, रिड्यूस टिलेज हो गया, इंटीग्रेटेड फार्मिंग हो गया, लाइवस्टॉक फार्मिंग हो गया, adaptation measures and international agreements, different international agreements होने चाहिए। एग्जांपल पेरिस एग्रीमेंट जो 2015 में किया गया था, इसमें हम क्लाइमेट चेंज के जो इफेक्ट्स हैं उसको मिटिगेट करने की कोशिश कर रहे थे इसमें। रूल्स एंड रेगुलेशंस, रूल्स और रेगुलेशंस इज मोस्ट इंपॉर्टेंट। डिफरेंट रूल्स बनाना चाहिए फॉर इंडस्ट्रीज, फॉर व्हीकल एमिशंस, जो कि क्लाइमेट चेंज को मिटिगेट करने में हेल्पफुल है। Public awareness and education, it is most important part for mitigating climate change. So there is no planet B, हमारे पास एक ही planet B है, एक ही planet है जिस पे हम रह सकते हैं, बाकी कोई planet रहने के लिए हमारे सर्वाइवल के लिए गुड नहीं है, इसलिए हमें अपने planet को सेव करना, बचाना है।
Thank you, Abdul Rahman. I would like to request Anupama Roy for the oral presentation as online.
Is it visible? The slides are visible? Or?
Yes, ma'am.
Okay. I hope I am in slideshow mode, right? Huh. Is it good? Okay. This is Anupama Roy. Wish you all a very good afternoon. Today I am going to discuss the topic on exploring the transcriptomic landscape of Ficus carica, a multi-tissue investigation. So our aim is to study the expression patterns across the Ficus carica, and why we are going to do, so that we can understand the activity level of genes in different tissues, developmental stages, and environmental condition. And it can help in the genetic improvement and breeding programs. So let's come to the introduction to the Ficus carica. It belongs to the family of Moraceae, and the common name is common fig or edible fig. And it have got the chromosome number of 13. and it is native to Mediterranean to Central Asia. And here in the right hand side, the botanical aspect of the Ficus carica have been shown, while on the left hand side, a graphical representation is shown. That is, area harvested is largest in Morocco with the 64,000 hectare, while the production is highest in Turkey with 320,000 tons. And if we talk about yield, it is 100 per gram hectare in unit, that is 251,000 in Colombia. And this all data have been fetched from FAO. So let's come to the genome statistics of Ficus carica. The first genome of Ficus carica, that is fig, that we call in Hindi as Anjeer, have been developed at the scaffold level for Horaceae cultivar with a sequence length of 247.1 MB, while the current chromosome level Ficus carica is used as DOTA2 with 333.4 MB, and the new in the year 2023, a new cultivar Moissoy Dauphine of Ficus carica have been developed with 375.2 MB. Let's come to the materials. I have taken care of the 12 bio project, all the 12 bio project that is available in the public domain, contributing to 64 samples. And I have categorized the 64 samples into 6 tissues, that is flower, leaf, peel, receptacle, syconium, and unknown. And I have used the reference genome for the Dorato Ficus carica with a genome size of 333.4 MB, and it has been developed sequenced by University of Pisa. Let's come to the methodology section. Methodology, we have retrieved the reads, FASTQ reads from the public domain repository. Then we have did the quality check using FastQC and the removal of the low quality reads and the adapter sequences on the reads present have been done using Thermomatic, and the reads have been aligned to the reference genome using HiSet2. The quantification of the gene expression level have been done using the string type tool, and all the transcript that we have got, we have calculated the coding potential, whether it's the coding or the non-coding, using the tool CPC2, and the gene ontology and pathway enrichment. This analysis have been done using BLAST2Go tool, while the R package TISPEC have been used for the tissue validation score, whether this gene is tissue specific or not. So this is the result section. We have come to total transcripts in the Ficus carica is 37,832. And here we have shown the non-coding transcripts is about 74.7%, while the coding transcript is 25.3%. And the coding transcripts serve as the template for the protein synthesis, while the non-coding transcripts act as a molecular switches, regulate the expression of the genes. Here is the result for the sample description, for example, for the syconium tissue, one of the tissue among the 6 tissues. And next come to the result section of the transcript information of the leaf tissue, to which chromosome it belongs, whether it's on the negative strand or the positive strand, which reference have been used, start and transcript length, peptide length, and the coding probability. And then it is the gene ontology for the leaf tissue. These are the GeoIDs, which represents to which molecular function, which biological process, and which cellular component it belongs, whether it is in the ribosome or in the Golgi body, anywhere. This is a very interesting thing, that is expression study in the form of heat map. So here I have showed the expression pattern of the leaf tissue with TPM, that is transcript per kilopase million. And in the section of the left-hand side, we can see A and B, the coding transcripts. And in the C and D, it's the non-coding transcripts. And the yellow side is showing the low expression value, while the red section is showing the high expression value. So more the expression level, more the intensity of the color. Here we have done a new thing, that is results in the tau index. Tau index is a measure of the tissue specificity score, which is given to each tissue, for each transcript in each tissue, and it ranges from 0 to 1. If the value is towards 0, it represents low specificity. If it is towards 1, it is representing high specificity. And in the right-hand side, I have shown the formula for calculation of the tau index, and I have done using TISPEC R package. So according to the score, there are 3 categories: housekeeping genes, that is come under 0.20, the tau score must be less than 0.20. And if it is more than 0.20 and less than 0.8, it is intermediate specific gene. And if it is higher than 0.8 and less than 1, then it is absolutely or highly specific genes. And this is the result in the middle, the teal color graph. It is showing the high or absolute specific gene, whether this amount, this 200 genes are in the flower are highly specific to flower only. And here with the orange, I have shown the intermediate specific gene, and in the blue color, it is non-specific or the housekeeping gene. This is the, for example, high tissue specificity gene in Syconium tissue, that is the fruit tissue of the pig. And it is the coding one with a tau score. As I have mentioned, if it is more than 0.8, then it is highly or absolutely specific. So let's come to the conclusion. I have used the state-of-the-art methods for the 6 different broad tissue categories, and I have done the gene ontology and the pathway analysis. The global expression pattern have been analyzed, and we have also made the categorization of the tissue-specific gene and the housekeeping genes that is involved in all the basic biological process of our organism. Future prospects, yeah, we need to do a web-based single platform that can aid the researchers to access and browse this collected data. We need to expand this work to every other horticulture as well as agronomically important crops. And by leveraging this gene expression data, improve agriculture productivity and resilience to environmental stresses can be done. So that's it. Thank you for your attention and patience. Thank you.
Thank you, Kumar. Now I would like to request Renu Kumar for your oral presentation is online.
टेबल पे एक्स्ट्रा ही रख दो।
Good afternoon, everyone. Hello. Is my slide visible?
Yes. Good afternoon, sir.
Good afternoon, everyone. My name is Vaibhav Kumar, PhD Bioinformatics. I am going to present on the topic long non-coding RNA subcellular localization prediction using multi-sources features. Coming to the introduction, more than 90% of the genome is transcribed, but later we found that only 1 to 2% of it is further translated into proteins and other remains non-functional. Different types of non-coding RNAs have been discovered in the last 2 decades, which have changed our understanding of the eukaryotic genome complexity and its functioning. Non-coding RNAs can be of long or short non-coding RNAs depending on its length. This diagram shows the distribution of long and short non-coding RNAs and the type of long non-coding RNAs. There are various functions performed by the long non-coding RNAs inside the cell, which can be seen in this diagram. The long non-coding RNA functions is very much dependent upon the subcellular localization of the long non-coding RNAs. It can be localized to the nucleus, cytoplasm, or any other cell organelles. We can see in this diagram the different type of functions performed by the long non-coding RNAs in the different locations inside the cell. Due to the increase in number of studies related to long non-coding RNAs, there is an urgent need of developing such a tool, machine learning based tools, to predict the subcellular localization of the long non-coding RNAs. So these are the general limitations of the tools which are existing right now for the subcellular localization of the long non-coding RNAs. So keeping in mind those gaps, these are the motivation for my present study. We can improve the feature set and improve the different machine learning and deep learning models. So this was the pipeline used in my current study, where data collection was made, then pre-processing was done, then a data set of 7 locations was collected. Then features were extracted, and then the data balancing was done to balance out the number of data sets in each of the classes. Then different machine learning models are training and tested, and then the best machine learning model was finally selected as our final predictor. These are the 7 locations considered in my study. The performance evaluation of the models will be done using the matrices based on the confusion matrix. Coming to the results, these were the number of data points in the 7 different classes. This diagram shows the data distribution of the 7 classes. Then the data balancing was done using SMOTE, which is synthetic minority oversampling technique. It creates synthetic data examples for the minority classes. These are the results for the combined features. The combined features were concatenated into one to get a large feature matrix. Then several feature selection methods like univariate selection, feature importance ranking, and recursive feature elimination was done to find the best set of features to train our model. These were the results obtained without the data balancing was done. As you can see, 62% best was found with random forest models. And this diagram shows the same different matrices using ROC curve and conclusion matrix. Then the best model was the random forest after the data balancing was done, which is 86%. This diagram also shows the same. So coming to the conclusion, we compared different feature set with and found that the best result was found with random forest. We trained and tested several machine learning models, and the hyperparameter optimization was done using grid search. Random forest was found best to be 65% with original data set and 86% with balanced data set. These are the references used in my study. Thank you.
Thank you very much, Kumar. Now I would like to request Dr. Harsh Manchanda for your oral presentation is online.
Good afternoon, everyone. Topic of my presentation is organic farming for sustainable agriculture. As we know, agriculture plays a crucial role in human growth and development in the industrialized and the emerging nations like India. And we know each and everyone is dependent on the agriculture for food and to earn living. But contemporary methods are generally used which adversely affect the agriculture, which adversely affect the environment. The use of pesticides is there, chemical fertilizers are there, and the high yielding are required.
Excuse me, ma'am, your voice is not clear.
Right. Now it's audible. Now is it audible? Hello?
Hello?
Yes, ma'am. Is it audible now?
No, ma'am. Now it is clear, ma'am.
Is it okay, ma'am? Now it's audible?
Yes, ma'am. Yes. Continue.
Okay. So in case of contemporary agriculture method, pesticides are used, chemical fertilizers are used, which adversely affect the environment. So there is a need for a practice which may be sustainable and is an eco-friendly manner. And one of these approaches is the organic farming. Organic farming, it is an environmentally responsible, creative and sustainable method. And the organic farming avoids the use of the inorganic farming. The chemical fertilizers are not used, the food additives are not used, and in organic farming, green manure, biofertilizers, and the mineral grade rock additives are used. According to the IFOM, International Federation of Organic Agriculture Movements, there are 4 basic principles of the organic farming. One is principle of health. According to this principle, organic farming should help to sustain and enhance the health of the soil, plants, animals, and the human beings. The second one is the principle of ecology. According to this principle, organic farming should be based on the living ecological systems and cycles. As the production is based on the ecological processes, it should help to maintain the ecological balance. And the third one is principle of fairness. According to this, organic farming should be built on relationships that ensure fairness with regard to the common environment and the life opportunities. And it should ensure fairness at all levels to farmers, workers, processors, and the distributors. And the last one is the principle of the care. According to this principle, that the organic farming should be practiced in such a manner that it protects the health and well-being of the current and the future generations and the environment. And for a crop to be considered organic, it should be grown in the areas free of the chemical fertilizers, synthetic pesticides, and herbicides at least for the 3 years prior to harvesting, along with a sufficient buffer zone to reduce the contamination from nearby farms. Areas for organic farming should be free from the sewage sludge, ionizing radiations, or genetically modified organisms. Methods for maintaining soil fertility include the crop rotation, use of the cover crops, use of organic fertilizers, and organic farming helps to make the system more resilient to the negative impact of the climate change and prevent soil erosion and resist the drought and change in the temperature, and help to improve the water holding capacity of the soil and maintain diversity of the microflora and fauna of the soil. Then organic farming also helps to cut down the release of the greenhouse gases like nitrous oxide and methane in the atmosphere. And organically grown vegetables, they are having the higher dry matter content, and the organic cereals, they are having high protein content. And the wheat, which is generally used as a staple food, it is having the 25 to 30% higher lysine content. Organically grown foods, they have 58% more polyphenolics than the conventionally grown foods. This may be due to the reason that plants have to face a large number of pests in absence of the synthetic fertilizer. So higher amount of polyphenols are synthesized, which act as the natural defense system of the organism. And when we talk about the sustainable developmental goals as a whole, agriculture either directly or indirectly contributes to each of the 17 sustainable goals. For example, if food security is not guaranteed, poverty cannot be eradicated. Enough food that is safe, affordable, and available is necessary to meet the second goal, that is zero hunger. Then the proper diet is the first step toward good health, and good health is required for learning and hence high quality education. Gender parity or gender equality can also be sorted because people in economically distressed nations are fed based on their gender. Water issue can also be resolved by sustainable agriculture. So to achieve sustainable developmental goals by 2030, agricultural methods need to be altered to ensure the supply of nutritious and enough food for everyone and to minimize the negative impact on the environment. Then there are certain constraints. Organic farming, in spite of the advantages, it is having the certain drawbacks like high cost and time commitments. In case of organic farming, high labor is required than the traditional agriculture method, and a farmer can produce more crops using conventional methods than with only the organic methods. In addition, lack of awareness, lack of quality manures, and marketing policies, inefficient agricultural policies, lack of financial support, and low yield are the other major drawbacks. So the techniques which can be used to overcome the constraints of organic farming are, as the organic fertilizers, they are not available in the sufficient quantity, a blend of the organic fertilizers and new technology is required to overcome the constraints of the organic farming. These are biochar, use of biochar, which is a pyrogenic carbon, a porous charcoal-like material obtained from the thermal degradation of the carbon-rich biomass in anaerobic conditions. Then the use of biofertilizers, which include free-living nitrogen-fixing bacteria, algae, and fungi. Then use of new technologies like nanobiostimulants and mycorrhizal fungi will also help to increase the agricultural productivity in an environment-friendly way. Then the use of sensor technologies like bee scanning app, which allows the beekeepers to combat the disruptor parasite and serve as a foundation for population modeling and breeding initiatives. And now finally we come to the conclusion. Organic farming, it is gaining popularity nowadays, but because of its healthier and safer practices and more safer and nutritious food, it is eco-friendly and is expanding at a very faster rate in the entire globe, including India. But still, there is a need to overcome the various weaknesses and threats to it, like the new pest and disease incidence to the organic crops, variability in the climatic patterns, and the weaknesses that the costly certification processes are there, time consuming, and weak government policies and lack of awareness in the farmers. Thank you.
Thank you, Dr. Harsh Matanda, for such a nice presentation. Now I will request to Dr. Vishwas Shankar Patil for oral presentation is online.
Good afternoon to all. Let me share my screen. Hi. Madam, can you share the PPT?
Yes, sir.
Please, ma'am, because PPT is not operating in my mobile.
Wait for some time.
Thank you, madam.
Yes.
Can I start?
Yes, sir.
Good afternoon to all. I'm Dr. Vishwas Patil from Satara, Maharashtra state. I'm going to present my research work entitled Studies on Phosphate Solubilizing Bacteria from Digester Effluent with reference to plant growth promotion. My presentation comes under the themes mentioned herewith, that is conservative agriculture, organic farming, etc. Next slide, please. Madam, next slide, please. Thank you, madam. Introduction. Phosphorus is the most vital nutrient required for plant and microbial growth. It is abundant in soil, but only 0.1% is available for plant uptake. Hence, farmers are usually applying chemical phosphatic fertilizers, but excess and inappropriate use has resulted into the environmental pollution, and also these chemical phosphatic fertilizers are costly. Alternative to these chemical fertilizers are biofertilizers, which are microbial preparations that are used to stimulate plant growth directly and indirectly. These phosphate solubilizing microorganisms are a type of biofertilizers that dissolve soil phosphorus and makes it available to the plants. Phosphate solubilizing bacteria have been isolated from different environmental sites, and their plant growth promotion studies have been performed by various researchers. Now see, biomethanation, which is a microbial technology, which is it is used to treat the waste, and the effluent that is generated is used as a manure. Now see, there are very few reports on isolation of phosphate solubilizing bacteria from digester, effluent, and hence the present study have been undertaken. Next slide, please. Material and methods. So initially, biomethanation of vegetable waste was performed by using 5-liter capacity locally fabricated digesters using operational and environmental parameters mentioned herewith. Then the digester effluent, it was collected and it was subjected for isolation of bacteria by using standard methodologies. Then the bacterial isolates that were obtained from digester effluent, they were subjected for phosphate solubilizing efficiency by qualitative means and quantitative means. Here for qualitative experiment, picoviscous media were used and hydrolyzing capacity was determined as per Hendricks et al. And for quantitative measurement, picoviscous broth was used after 10 days, the standard graph of KH2PO4 was taken into account for determining the amount of released phosphorus by each isolate. Then the potted isolates, they were selected further to produce whether they produce other plant growth promoting parameters like indole acetic acid or indole acetic acid, hydrogen cyanide, then ammonia, siderophores, etc. Then these two bacterial isolates, they were subjected for determining plant growth promotion studies by using pot assay method. And here 4 pots were used. C1 labeled pot contain only sterile soil. C2 labeled pot contain sterile soil plus tricalcium phosphate. Then B1 labeled pot contained sterile soil plus tricalcium phosphate plus B1 culture that was coated over the seeds that were sowed into the pots. And B2, that is fourth pot, it contains sterile soil plus tricalcium phosphate plus the B2 bacterial culture, which was used to coat the seeds of the plants. And here, these 2 isolates were used to study plant growth promotion in using maize, jowar, and wheat plants. And then these bacterial cultures, they were identified by using standard methodology. Next slide, please. Results and discussion. So biomethanation of vegetable waste. Here, the biogas yield is represented here. The digester, it was used to isolate bacteria. And here 17 bacteria were isolated and stored in refrigeration conditions. And they were further screened for determining their phosphate solubilizing efficiency. And based on hydrolyzing capacity and quantitative studies, B1 and B2, these 2 most potent bacterial isolates, they were used for further studies. And these 2 isolates, they were also found to be producing indole acetic acid, siderophores, ammonia, hydrogen cyanide, and catalase as well. Then these 2 isolates were used for pot study. Next slide, please. These are the results of pot studies for effect of inoculation of B1 and B2 PSB isolates on the growth of maize plants. Figure 2 represents effect of these 2 isolates on shoot and root development. And here table 1 represents the data for shoot length, root length, shoot fresh weight, dry weight, root fresh weight, root dry weight in response to C1, C2, B1, and B2 treatments. Next slide, please. These are the pot study experiment for effect of these 2 isolates on the growth of jowar. Figure 3 and 4 represents the effect on shoot and root development. And here again, table 2 represents the shoot and root parameters of jowar in response to C1, C2. B2 treatments. Next slide, please. So this is the third plant that is used, which is a wheat. And here again, the figures represents the pot studies for effect of B1 and B2 isolate on shoot and root development in case of wheat plant. Next slide, please. Here, please note down these 2 bacterial isolates that were used to study plant growth of maize stimulated shoot development by 8 to 18% and root development by 2 to 12%. Then in case of jowar, B1 and B2 isolates stimulated shoot development in the range 15 to 92% and root development in the range 3 to 173%. And in case of wheat, both isolates didn't stimulated growth significantly. And here, these tables that I showed here, the statistical analysis is done And in statistical analysis, it is found that B1 and B2 stimulated maize and jowar growth significantly. And among these two isolates, B2 was having more plant growth promotion as compared to B1 isolate. Next slide, please. Next slide, please. These are the graphical presentation of effect of these two isolates on shoot and root development in case of maize, jowar, and wheat. Next slide, please. Now see, these two isolates, B1 and B2, they were identified by morphological, cultural, and biochemical characterization, and their identity was confirmed by molecular analysis, such as 16S gene analysis, and B1 was found to be Bacillus amyloliquefaciens, and B2 was identified as Brevandimonas diminuta. Next slide, please. Conclusion. Here, the present study showed that these isolates can be used as biofertilizers for sustainable development of agriculture by working as a substitute and reducing the use of chemical fertilizers. We are still using the cattle dung, that is a digester effluent of a biomethanation plant, as a manure. But specifically, I have tried here to isolate the PSPs, and I have exact findings. Based on these findings, it can be used as a biofertilizer. Next slide, please. These are the acknowledgement. Next slide includes the references. And lastly, I thank you for being patient, and for sorry for a technical error. It is in PPT, so I can't open it in a Zoom manner. Thank you, madam. Thank you for providing my presentation. Thank you, madam.
Thank you, Dr. Vishwas Chandra Patil, for your nice presentation. Now,
Thank you.
request to Dr. V. Rushali Ashok Wagh for your oral presentation is online.
Yes, good afternoon, ma'am. Just a minute, ma'am. I am sharing my presentation.
Finish your within 5 minutes. Presentation within 5
Ma'am,
minutes.
can you share?
Yes, ma'am.
उसके बाद ये लास्ट है रिसेंटली बंद हो गया ना बस ये शांत रहो कल्चरल स्टार्ट हो ठीक है चाय आ रही है इधर ही बिना चीनी वाली आई है ना सबकी चाय आ रही है
आज आप नीचे आ जाओ
9:45 कर ले 9:30 का बच्चे 9:30 कर दो अपने बच्चे बिठा दो फिर हां 9:30
Thank you, ma'am. Can I start presentation?
Complete your speech within 5 minutes.
Yeah, definitely, ma'am. I will complete. Yes, my presentation topic is degradation of imidacloprid and its metabolite imidacloprid olefin by fungal isolate and its applicability for soil bioremediation. Next slide, please, ma'am. All of us know that most of the farmers are actually using chemical fertilizer, but there are the number of side effects of chemical fertilizers which are responsible to decrease the fertility of the soil, which are harmful for the human, for the animal, and for the environmental. So yes, next slide, ma'am. For that purpose, we selected one process, that is the bioremediation process. Yes, because this bioremediation process is not costly one. It is not responsible to form any toxic metabolite. It is eco-friendly, versatile one. And microorganisms means we selected fungi because fungi have the ability to form extended mycelial network. They have the low specificity for their catabolic enzymes also. And yes, the most important is fungi are critical to the biogeochemical cycle, and through the toxification and mineralization, these fungi have the ability to convert toxic substances into the eco-friendly substances, means non-toxic substances. Ma'am, next slide. And our targeted pesticide is the imidacloprid, which is neonicotinoid pesticide. Farmers are generally use this pesticide to control the number of a pest which are generally attacking on the vegetables also, then on the sugarcane also, etc. And it is one of the harmful pesticide. Ma'am, next slide. Yes, these all are the characteristics. Ma'am, next slide. Yes, this why we are selecting imidacloprid? Because it have the long shelf life, half-life, which varies in between the 26 to 229 days. Means during this period, it is totally responsible to damage the soil. It is responsible to show the various toxic effects, and not only imidacloprid is toxic, its metabolites are more toxic than the imidacloprid. Half-life is more. That's why we are selecting this pesticide. And in North Gujarat region, the number of the farmers, and in Maharashtra also, number of farmers are using this pesticide. Next slide. And these are our objectives. Yes, properties of the imidacloprid. This is the structure of the imidacloprid. Molecular weight is 256 gram per mole. It is water soluble. Melting point is 136 to 143 degrees Celsius. Next slide. Yes, these are our aims and objectives. Yes, these are the different metabolites which are formed by the imidacloprid, and these are also the toxic one. You can see imidacloprid olefin, which is our targeted metabolite. Second one is the urea. All these are also the very harmful. These are responsible to cause the groundwater pollution also. Next slide, ma'am. Yes, these are our objective. First one is the enrichment isolation, then the screening, then the optimization of media components by using the statistical analysis like the Plackett-Berman design and the CCD. And next one is the bioremediation study. Next slide. For that purpose, we collected the soil samples from the field in which the farmers are continuously using this imidacloprid pesticide, because they are only the resistant microorganism which resist the high concentration of the imidacloprid. These are present. Next one. Then we perform the enrichment. Next slide. Yes, enrichment was carried out by indigenously fabricated column. In this particular design, we are taking the soil sample from the field and continuously we are applying the pesticide, different concentrations of the imidacloprid, you can see. And due to that, we can check the resistance of the microorganisms also towards the imidacloprid. You can, at the right side, you can see microscopic observation of the fungi. And yes, isolation was done by the serial dilution method, and the media which is favorable for the fungi, we selected Zapadox media. Next one. After enrichment and isolation, we check the screening, means we check the tolerance. You can see these fungal isolates tolerated a high concentration up to the 1 lakh ppm also. Next slide. After completion of the gradient plate, we perform the MIC also. At MIC, you can see there is the one fungal isolate which tolerated the high concentration, and that is the F1 fungal isolate, which tolerated up to the 1 lakh ppm or 1 lakh mg per liter concentration of the imidacloprid. Yes, ma'am, please next slide. After selecting the favorable fungi, then the extraction is carried out. Yes, we are taking the concentration of the imidacloprid, that is 100 mg per liter, and you can see by using ethyl acetate, we extracted the imidacloprid residues at the upper side, organic phase. At the lower side, there is the aqueous phase. Next slide, please. Next. Yes, by taking these 3 isolate which are tolerating high concentration, we perform the growth kinetic study and the degradation kinetic study. You can see this is the degradation kinetic study. Initially, we discussed that half-life is 2 more, up to 200 days. But when we are using this F1 isolate, the half-life get decreased, and that is reached up to only 27.72 hour. Here we perform the degradation kinetic study and the concentration was 100 in within the 5 days. That decreases up to the 0 mg per liter. Okay, next one. Next. Yes, ma'am. Going fast now. Now this is the growth kinetic study. Initially, organism is not tolerated the concentration, but after adopting the pesticide as a carbon source, phosphorus source, then the growth of the fungi get increased in presence of the high concentration of the imidacloprid. Next slide, ma'am. Sequencing was carried out by using 18S rRNA sequencing, ma'am. Next slide. On the basis of 18S rRNA sequencing as concerned to the phylogenetic tree. Okay, this fungi is responsible to degrade the maximum amount of the pesticide. Next one. This is the phylogenetic tree. Then we check the acclimatization study. In that we lack all the carbon source, nitrogen, phosphorus. Still, the fungi have the ability to grow and to degrade the imidacloprid. This proves that it is utilizing pesticide as a sole carbon source. Ma'am, next one. Toxicity was checked by the Allium cepa assay. You can see in the biodegraded media. Ma'am, next slide, please. In biodegraded media, we can see the different phases. Yes, this is the seed germination assay. Here also, the high concentration of the imidacloprid is affects on the growth of these seeds. Okay, ma'am, next slide. The next one, Allium cepa assay was carried out. These are the images of the control. Next one, next slide, in which we are taking the biodegraded media. Means this result proves that the no toxic metabolite is formed and the overall process is the eco-friendly one. Ma'am, slide is not visible. Hello. Ma'am, slide is not visible.
Ma'am, time is over.